Good morning, ladies and gentlemen. It's my very great pleasure to welcome you to the 19th Vienna Congress. My name is Evelyn Baer, and I will be your host for the upcoming 2 days. You are going to have a brilliant Congress, and you will take home a lot with you today and tomorrow. The Vienna Congress is a think tank where the most pressing issues of the world are being discussed. After 2 years of uncertainty and global turmoil due to COVID-19, the world is slowly emerging from the pandemic. With life returning to normal ever so slowly, we now dare to look ahead and discuss the future. Hence, this year's Vienna Congress focuses on strategies for a sustainable upswing. Top-class international, European and Austrian decision makers from business, science and politics discuss scenarios for the world ahead. As a result, at the end of the Vienna Congress, the Vienna Declaration will be published. It's the 19th Vienna Congress, and we are streaming live into the world. We'll obviously have some speakers joining us online, but we are delighted that most of our speakers join us here in Vienna again. Of course, we comply with all the strict security measures. Now I have one request for today's speakers. I would like to ask you to be mindful of timing during your speech, because we have a lot on our agenda today. Last but not least, I would like to thank all of our sponsors for today and tomorrow. So now let's begin with 4 short opening statements. Please now welcome on stage Dan Schechtman, Nobel Laureate in Chemistry and Vice Chair of the Advisory Board of the Vienna Congress.
Ladies and gentlemen, it's a pleasure to be here for, I don't know how many times I've visited here, and it's always a great event and a memorable event. Congratulations to the amazing David Ungerklein for holding the Vienna Congress even in these challenging times. The Congress brings together top-class speakers and highly relevant pertinent topics, and above all, about the corona pandemic. Today, it is more important than ever to convey scientific basis, for example, about vaccinations. Not everybody is an expert in this, and in this meeting, we'll hear a lot about vaccinations. The Congress of Vienna sends a strong signal for scientific achievements, and they're important for the future of the world. Science, research, and innovations are becoming increasingly important for human and the economy of the world. Hostility to science is ultimately hostility to the future of our countries. That is why we have to encourage curiosity, critical questioning from a very early age. It's times like this, the Vienna Congress asks the right questions about health, the economy, economic recovery, regions, and the climate. It's up to all of us to give and implement answers that will save our future and make it safe. Thank you.
Thank you very much, Dan Schechtman. Please now welcome on stage Alfred Hahl, Chairman of the Austrian Professional Association of Management Consultancy, Accounting and IT at the Austrian Federal Economic Chamber.
Good morning, ladies and gentlemen, Mr. President, cabinet members, and my colleagues from the industry of management consulting. I'm honored to share my perspective again with you on this extraordinary Vienna Congress today. We want to find out strategies for a sustainable upswing and to save the future. David Ungerklein organizes this event again, and especially in the pandemic year. And not even Corona could stop him. This requires courage, foresight, and an incredible amount of work. The reward is that David brought leaders and independent thinkers to the Vienna Congress. The pandemic is not yet behind us, but we must learn to deal with it and turn our attention to the essential issues of the future. We have many options, but strategy for the future are digital and are known. The top trend that promises the most in the short term is still artificial intelligence. This is because, according to McKinsey, artificial intelligence combines the possibility of being used in all sectors and industries, with the majority of the technology. Artificial intelligence is no longer merely a topic in universities, but has reached market maturity in many places. No other technology combines the potential to bring disruptive change with the maturity to be deployed now like artificial intelligence. Artificial intelligence can recognize patterns and make decisions where humans have long since exceeded their limits. Artificial intelligence is already being used in both manufacturing and at the point of contact with customers. McKinsey predicts that as early as 2024, half of customer interactions will be artificial intelligence based. It has so many applications that they will be absolutely essential in the future. Let me give you a current example from the field of medicine to underline the benefits. Artificial intelligence achieving high performance in diagnosis, prognosis evaluation, epidemic prediction, and drug discovery for COVID-19. AI has the potential to enhance significantly existing medical and healthcare system efficiency during the COVID-19 pandemic. The World Health Organization signaled that it could be an important technology to manage the crisis by the virus. It's a core technology of the fourth industrial revolution, is an important non-medical intervention to overcome the current global health crisis, to build next-generation epidemic preparedness, and to move towards a resilient recovery. But to make digitalization and artificial intelligence possible, in the first place, the right experts are needed in addition to the appropriate infrastructure. And this is where the problem begins. Let me share some facts with you. According to the Digital Economy and Society Index 2020, the advanced skills and developed subdimension looks at the workforce and its potential to work in and develop the digital economy. This takes into account the percentage of people in the workforce with ICT specialist skills, with a breakdown for female ICT specialists. At the same time, it looks at the share of ICT graduates. Following the digital decade communication, the key target for Europe is to reach 20 million employed ICT specialists in the EU by 2030, with conversion between women and men. In 2020, nowadays, just 8.4 million persons worked as ICT specialists across the European Union. The share of ICT specialists is slowly progressing and reached 4.3% of total employment in 2020. The majority of ICT specialists in the EU are with 82% men in 2020. Since we are in Vienna today, please allow me to share with you what it looks like in Austria when it comes to employment. Last year, Austria lacks over 24,000 IT experts, and the number is increasing every year. By the way, Germany lacks over 94,000 IT experts. All over Europe, about 11 million additional IT experts are needed in the next 8 years. Dear ladies and gentlemen, 11 million, that's almost the population of Sweden. That's why I'm asking governments at national and European level to provide universities with the necessary tools, but also to take vocational schools and teaching into account accordingly. Otherwise, the IT light will go out in Europe of a lack of experts. If the EU wants to compete on the global stage, ladies and gentlemen, there is no time to be lost. We need to act right away. We must have plans for all European member states that show how we can produce 11 million IT experts in the next 8 years. But that is only a basic requirement and not a guarantee of success. However, one thing is for sure, consultants are more important than ever before. I hope you enjoyed my insights and that the outcome of this creative day will assist our government and will take Austria to the top of Europe and make Europe a major player in the world. Dear David, congratulations. We need more leaders like you so that we are front runners, not only Austria, also Europe in the next 8 years to get 8 million or more IT experts. Many thanks.
Thank you, Alfred Harl. I'd now like to introduce Thomas Seckeres, President of the Austrian Medical Association and the Medical Association of Vienna.
Dear ladies and gentlemen, the Vienna Congress places a strong and valuable focus on health, science, and Corona. It is important to contrast the rampant hostility to science with the facts and context. There is a lot of persuasion ahead of us, and I'm very pleased that the Vienna Congress has succeeded to bringing the world's top representatives of the medical profession to Vienna, the president, former president, and Secretary General of the World Medical Association. This is a very strong signal for Vienna as a health location. This is a strong signal for our doctors. I'm also very pleased that the Vienna Congress is doing the groundwork on the subject of vaccination, because vaccination will play an even more important role in the future of medicine. And it is a great honor for me that the biochemist Professor Katalin Kariko is taking part in the Vienna Congress. Her research work is the groundwork for the development of mRNA-based COVID-19 vaccines that have now been used many billion times. For us doctors who have been and are at the forefront of the fight against Corona worldwide, vaccination technology is the key technology to overcome this pandemic situation. I would also like to take this opportunity to thank those who deliver extraordinary performances here in hospitals and outpatient clinics. And I would like to remind you all of the many patients and doctors and healthcare workers who lost their lives in the fight against Corona. Save the Future, therefore, a particularly accurate title for what we are going to discuss here today. It is not just about economic figures, it is about personal existences and fates. This is our mission as doctors, and for this mission we need the best possible framework condition. I wish you all an exciting and interesting Congress, and I would like to thank the organizers, in particular David, for organizing this important meeting. Thank you very much.
Thank you, Thomas Seckeres. And now finally, let's welcome David Unger-Klein, the founder and initiator of the Vienna Congress com.sult.
Dear guests, ladies and gentlemen, distinguished President Václav Klaus and Nobel laureate Dan Shechtman, who are both chairing the Vienna Congress for many years, I would like to welcome our members of the advisory board. Welcome to our Vienna Congress 2022. 57 speakers on 2 Congress days from 3 continents, 5 Nobel laureates, former presidents, numerous secretary generals of well-known institutions, and a personality who was and is named Hero of the Year 2021 on the cover of the December issue of the Times magazine. It is the second hybrid Vienna Congress, but it does not detract from our ambition. On the contrary, last year we were able to attract around 32,500 views worldwide in these 2 days. This year's main topic is Save the Future, strategies for a sustainable upswing. And with good reason. There are many areas where we have to deal more and better with our future if we want to have a good future. We will therefore debate the health, economic and democratic policy challenges of the corona crisis today and tomorrow, and show what we have to do for recovery and ways out of the crisis. I'm very, very pleased that we were able to attract top-class speakers from all over the globe, above all, Professor Katalin Kariko. From a research perspective, she's the mother of the mRNA vaccines that are currently being vaccinated more than 8 billion times, very successfully against corona worldwide. 5 Nobel laureates, a number of scientists and decision makers from economics, politics, and institutions will participate in our deliberations. Thank you all for your valuable impulse. At the Vienna Congress, one thing is traditionally particularly important for us. Building bridges between different disciplines and departments, countries and regions, views and opinions. This is particularly important when it comes to saving the future, because the future can only be shaped better together. The corona debate in particular shows that the future needs more bridge builders. That's why we promote discourse between top scientists, decision makers, and also critical voices at the highest level, and broadcast these deliberations worldwide via live stream. The future comes from working together and bridging gaps, and not from splitting and dividing. With this in mind, ladies and gentlemen, I wish you 2 exciting days at the 90th Vienna Congress and many good perspectives for the future. Thank you.
Thank you, David Unger-Klein. Our first topic today focuses on the importance of science. First of all, we will hear 2 keynote speeches and 2 introductory notes that will get us in the mood for the following panel discussion on a world between scientism and an anti-scientific worldview. It's my pleasure to now welcome Eva Condoroschi, recipient of the Balsam Prize 2018 for Chemical Ecology, member of the group of Chief Scientific Advisors of the European Commission. She will introduce our first keynote speaker of the day, Harald Zuhausen, Nobel laureate in physiology or medicine.
Thank you very much. Ladies and gentlemen, we have the privilege today to have in this morning session scientists who changed the world by their discoveries. I am very pleased to introduce you Harald Zuhausen, Nobel laureate in medicine. Professor Zurhausen is a virologist born in Germany who received the Nobel Prize in 2008 for his discovery that cervical cancer is caused by human papilloma viruses. Cervical cancer is a deadly illness, and this is the second most frequent cancer in females. He identified HPV 16 and 18, the most aggressive viruses, and made it available for the scientific community. Vaccines against HPV viruses can protect against the infections. And now this is the way how to detect the best way the cervical cancer. HPV dissection and vaccination against HPV will make really the change in the future. In the European Commission, in the group of chief scientific advisors, we are working on recommendation on new cancer screening programs. And one of these recommendation to advise for the vaccination for girls at the age of 15 or below to get the vaccination, because this can lead to the eradication of the human papillomavirus infection and thereby the cervical cancer. So this is a great privilege for me to meet Professor Zurhausen, and I would like to give you the floor now for your talk. Thank you.
Well, first of all, thank you very much for the very kind invitation to come here to this meeting. For me, it's the first time to participate in this meeting, but I understand it has been very successful in the past year. So, as an initial address, I'll try today to introduce you to a novel class of infectious agents and to try to discuss with you the link to chronic neurological diseases, specifically mainly to multiple sclerosis, subsequently to very common human cancers, in particular cancer of the colon, cancer of the breast, and cancer of the prostate. And subsequently, I'll turn in the end to a very interesting, the most common disease in humans here, namely to diabetes mellitus type 2, which we believe has also an infectious origin as well. Now, we know at present a number of infectious agents which we call direct infectious carcinogens. Direct carcinogens means that they persist, genomes or part of the genomes persist in tumor cells, are expressed in tumor cells, and only under those conditions the cell will turn eventually to a malignant cell. A couple of them are listed here. For instance, polyomaviruses, Epstein-Barr virus, papillomaviruses, and a few others. Now, the most remarkable feature here is that they require the persistence of specific pieces of those genes, of the total genome, in order to maintain the malignant phenotype of the respective types of cells. This is quite different from another group of infectious agents, namely those which we like to call indirect carcinogens, which are clearly linked to specific cancers, like Helicobacter pylori, a bacterial product which is found in gastric cancer, like the couple of parasitic infections, liver flukes, and also a couple of others, which cause chronic infections, particularly in tropical countries. Here, one important other disease is hepatitis C virus, which is not persisting in the cancer cells, but causing chronic inflammatory lesions in the liver, and which leads eventually to liver cancer. And then the so-called human immunodeficiency viruses, 2 types in the meantime, which lead to immunosuppression, where immunosuppression does not require the persistence of those agents in the cancer cells, but leads to the increase of certain other types of cancer, which I'm going to discuss with you in a moment. And then there's another class, and a new class of agents, which we will discuss today in more detail, which we like to call bovine milk and meat factors, a rather peculiar class, which are not representatives of viruses or bacteria, but something different. I'll try to discuss it with you in more detail. So we know at the moment different modes of contribution of infections to human cancer development specifically, namely, as I said before, the direct carcinogens, where you require the insertion of total or pieces of specific genes into infected cells, the persistence and gene expression in those types of cells, and which results eventually, even under experimental conditions, to so-called immortalization, to continuous growth in tissue culture, or subsequently even to malignant conversion. And the second group are those indirect carcinogens which induce immunosuppression, and this is best studied, of course, in the case of human immunodeficiency viruses. And finally, another type of indirect carcinogens, and they are the ones which I will discuss mainly, chronic inflammation inducing, which leads to the formation of oxygen radicals, the random mutagenesis in replicating adjacent cells in various types of tissues, and they are relatively poorly and only have been recently being studied to some degree. Now here we have a chronic inflammation due to specific infections around the replicating tumor progenitor cells, not in the tumor cells, in the tumor progenitor cells themselves. The oxygen radical production inducing mutations in adjacent proliferating cells. These are random mutations in the genomes of the replicating also in the genomes of the replicating infectious agents. And finally, cancer occurs after random hits in so-called driver genes, driver mutations, which happen and which lead to cancer here in these cases, 40 to 70 years after primary infection. So an enormously long latency period unknown in previous examples. I try to summarize here the mechanism by which they contribute. I will only stress the main point. So in direct carcinogenesis, infectious carcinogenesis, we can state the malignant growth depends on the specific expression of these persisting viral genes, and an increased frequency of those cancers occurs under conditions of immunosuppression, for almost all of them. But for indirect carcinogens, the situation is quite different. Namely, there's no persistence in the induced tumor cells or even in the precursor cells of those types of tumors. And malignant growth results from the induction of specific inflammations with the production of oxygen radicals, which induce random mutations. And the random character of those mutations explains the long latency period for the cancer development, because specific genes have to be hit under those circumstances. Now, well, we started out from epidemiological situations which pointed to us that maybe in view of the literature which was published, that in red meat there are some things which happen commonly in the world, namely that the consumption of red meat is a risk factor for certain types of cancers, in particular cancers of the colon, also to some degree cancers of the breast and a couple of other cancers. And that triggered the idea initially that we have to look for something which might be infectious because it was localized to specific countries. In one country which eats a lot of red meat, for instance Mongolia, in all kinds of preparations, the rate of these types of cancer is amazingly low. But in others, Europe, United States, and a couple of other countries, it's just the opposite situation. The rate is very high. So is it possible that it is due to a specific type of red meat which is consumed? So we suspected it, and according to the investigations which we did at that time, it came up that it is probably mainly the beef, the meat of our Eurasian type of cattle, which is responsible here. And so for that reason, we started very early, based on work which Esther Michel, who is here in this audience, and who is really the member here, she is the joint presentation person here today, in a way, to study initially single-stranded small DNA viruses, which we are, all of you who are sitting here probably carry them for a lifetime in your peripheral blood. And we had the initial suspicion based on studies on Epstein-Barr virus, another different agent, that persistent infection after long periods of time may lead to some malignant diseases. As the outcome of these studies didn't lead to anything. We couldn't find anything which was linked to cancer in this respect. But the methodology which has been developed there was very helpful in analyzing material which we suspected to be important, namely, in particular, dairy material, from dairy cattle milk materials, and milk products, and also meat products in the peripheral blood. So we started with the help of a veterinary institute in Leipzig, Professor Müller, to analyze 130 sera from healthy cows to see whether something which is suspicious there in this respect. And Esther Michel has been, my wife here, is quite successful in isolating a number of single-stranded molecules, circular molecules, initially with the tendency to isolate TT viruses, which are so frequent in humans, but they were different from TT viruses. They showed us the molecular structure after sequencing the DNA of them. That's quite different from them. And there were a number of types which came up, or genotypes, which came up very early. I will only discuss two of them because they turn out to be important, which we call bovine milk and meat factors. I'll come to this in a moment. They were related to an isolate which was initially obtained by Laura Manuelidis in Yale from a transmissible spongiform encephalopathy, the mad disease of cows, and also scrapie of sheep. And, but still, they showed some interesting differences. They are small molecules, 1,760 bases, or about 360 bases, but they turned out to be very interesting. Now, the most interesting part, which came up already instantly when we isolated those from the peripheral blood of those cattle, which we received from Leipzig quite regularly, that we found many of them in the milk products, in the supermarkets around Heidelberg. I'm afraid the same accounts for Vienna as well, and many other places in the world. They are present there. They are in yogurt, they are in creme fraiche, and we have not found them in cheese and butter, but it doesn't exclude that they're presently there. So our initial suspicion was that these are specific products of our dairy cattle, the Eurasian type of aurochs-derived cattle, which turned out to be wrong, this assumption, because recently a group in Munich who was asked by the milk industry, who received a number of requests to analyze the annoying types of statements from our group that milk might contain something infectious, to test this, our hypothesis. And this group came up with rather neat confirmation of our own results, and in addition found it in water buffaloes, also sheep and goat milks as well. So this is a kind of a bovine type of infection which we can characterize right now, although we cannot fully exclude that other species may be affected as well. Well, Esther Michel did a very careful analysis of all the genomes which we have isolated, sequenced, and deposited. They're even patented right now, which here composed of two groups, which we call BMF1 and BMF2. BMF for bovine meat and milk and meat factors. And you see here about, 20 BMF1 types and about, 110 or something like this of BMF2 types, which I think it's similar to papillomaviruses. The number may increase in the future as well. But what are the characteristics of these types? They are neither viruses nor bacteria, but they are related to specific bacterial plasmids, so the mini chromosomes of the bacteria. And these here are specifically related to one bacterium, namely Acinetobacter baumannii, where these plasmids are being found. That's cryptic plasmids there. Most of them are acquired by bovine dairy products and by meat consumption. And we propose as a name right now, and as you can see on the particle structure on the upper right-hand picture of this building, we could demonstrate them with monoclonal antibodies, which we developed in our lab, as kind of a pleomorphic type of structure. They're neither a typical viral structure nor anything typical for bacteria. And we propose to name them here as pleomorphic infectious vesicles. Even in some instances, it appears that have these vesicles fused with each other, so they are larger than one individual's vesicle. But the nanogold particles here coated with the monoclonal antibodies against a specific antigen of those agents permitted us to identify them. Otherwise, in the electron microscope, we would have never identified them, as otherwise it would have been typically exosomes or something unspecific there, but they are quite specific. So, we presently guess that these are the most common zoonosis as diseases transmitted from animals to humans. And what I will try to develop to you, that they are linked to probably at least 50% of the global cancer incidence in humans, and they're also involved in certain chronic neurological diseases, and as I will discuss at the end, in the age-dependent type 2 diabetes mellitus, the most frequent common disease in humans. We call them all indirect carcinogens. They are not persistent in the cancer cells. We try to find their genomes in tissue culture cells from colon cancer and others and fail to do so. But they are not affected and not induced after immunosuppression. It's just the opposite is taking place here, which is quite different to infectious agents caused by direct carcinogens, namely they are reduced. They are reduced under those conditions. Immunosuppression has a protective effect for these types of infections, in particular HIV infections or organ transplant recipients have a reduced incidence of colon, breast, and prostate cancers, those which I will discuss mainly today. And in combination with infected macrophages, namely in particular in the colon for CD68 positive macrophages, they commonly induce chronic inflammations, which go on for years and decades, and induce chronic mutations in this period of time due to the resulting oxygen radical synthesis, which can be demonstrated. The induction of mutations in DNA synthesizing cells, which are adjacent to the infected ones, are really the ones which are the cancer progenitor cells and lead to polyfusion induction in the case of colon hits and later on to colon cancer. They require several hits in addition to different genes of the same cell. The same cell has to acquire several hits in several specific types of genes. That explains for the likelihood of these events the long latency periods of those diseases linked to them. And the infection of mutations in DNA synthesizing as adjacent cells in case is really due to the effect, has really the effect of cancer induction in the end, requiring several hits in different driver genes. And as I said before, explaining the latency periods. Why did we name them bovine milk and meat factors, or BMMFs? Because the majority of them have been isolated in milk and dairy products, initially from Eurasian type of cows, subsequently by a group in Munich who was asked by the milk industry in Germany, who was annoyed by the requests from the public, from our publications, that the milk contains something which maybe in the end, or for more decades dangerous for humans, to reproduce our findings or to prove that they are wrong. Unfortunately for the milk industries, it turned out the opposite. They could confirm completely our finding at the Microbiology Institute of the Veterinarian Faculty in the University of Munich. And they confirmed them and found even the same agents or similar agents in water buffaloes, sheep, and goats as well. So they emerge as human infections after transmission from milk and bovine meat products. They cause chronic inflammation, the oxygen radical synthesis leads to the formation of 8-hydroxyguanosine as a sign for the action of oxygen radicals, and often this occurs after decade-long periods. Mutations in the adjacent replicating cells are then the rule. After mutation modifications in commonly more than one driver gene, this comes to the development of precursor lesions in the colon, for instance, to colon polyps, and cancer after long decades latency periods, as I said before. The frequent and almost ubiquitous infection of humans exposed to this food cries really for a need of preventive interferences. Since the vast majority of humans, and I'm afraid probably all of you sitting here in this moment, consume during lifetime, particularly in an early period of life, contaminated bovine milk and meat products, the BMMF ubiquity in humans, and there is really ubiquity, points to the importance as by far most frequent zoonosis in humans. So, which are identified up to now if you disregard malarial infections which are transmitted by mosquitoes, which are probably as frequent, at least in general. What I'm going to do today is to discuss with you three types of, we call them phases of BMMF infection. And one of them will be chronic neurological diseases. And I will use here as an example the multiple sclerosis, because the first two BMMF isolates in the slide which I showed you before were also obtained from one autopsy material of a patient with multiple sclerosis. And from this one patient, Ethel Michel could isolate two distinct types of BMMFs, which are, as we see today, at least one of them is clearly the most prevalent type, which we have found at least in colon cancer as well. And secondly, we postulated that multiple sclerosis originates from a dual infection of the same cell by two different infection factors, which lead, that we studied in the past quite intensively. If herpes viruses like Epstein-Barr virus infect a cell, and the same cell contains genomes of small circular DNA, a huge amplification of the small circular DNA, and an inhibition really of the induced virus which leads to this type of amplification. And so we have a kind of example where the integration of the EBV plays, or the presence of the Epstein-Barr virus in these types of infections plays an important role, namely leading to the amplification of a second factor, which has been also postulated in a recent publication which went through the press by Ascario and his group in Boston, who claimed there must be a second factor. We believe that we know the second factor, which exists here. It's a model with integration of the EBV role, a second infectious factor, DNA amplification of the second factor, chronic inflammatory response, and in addition, recent data show that the serology is easily compatible with this type of model. I will discuss it a little bit more in detail in a moment. I will also discuss as another example for indirect carcinogenesis, colon cancer and pancreatic cancer as well. And then subsequently, type 2 diabetes mellitus. I think there's an error there. It's 2015. 14 is a wrong statement. It's 2021-22. There's a BMF infection, which I will discuss when I come to this topic. We developed a model of multiple sclerosis which combines all these factors which I just mentioned briefly before, namely that we have two preconditions for the development of multiple sclerosis. One is the rare happening of a dual infection of an individual cell, usually in venous caves in the brain, by bovine meat and milk factors and Epstein-Barr virus, leading to a latent infection in such kind of cells. And secondly, a precondition is a kind of a vitamin D deficiency. Vitamin D deficiency, as we knew from studies which were conducted years ago, particularly before I came to Heidelberg, it leads to an activation of TGF-beta, tumor growth factor beta, and this tumor growth factor beta has a very interesting property, namely it leads to the induction of Epstein-Barr virus from a latent state to an active activation of the virus itself. So what happens here, if these two preconditions exist, the excretion of the production of an activation of Epstein-Barr virus, and suddenly at the same time a huge amplification of the BMMF infections, single-stranded small DNA, which is amplified highly under those conditions. And this highly amplified DNA, according to our hypothesis, leads indeed to the infection of the neighboring cells, to the very active inflammatory response, which is shown on the lower left part of this picture, and to the so-called autoimmune response, which is, in our view, not really an autoimmune response, but an immune response reacting to this type of specific induction in those types of cases. Well, we could demonstrate that the amplification of similar agents like our BMMFs, in the case of Epstein-Barr virus, as Michel did it with our group, demonstrating that under those conditions, indeed, TT viruses, which are similar genomic structure, but as far as nucleic acid composition is concerned, very different, it leads indeed to a rather enormous amplification of TT virus DNA under those circumstances. So at this moment, the model here, which I present to you, is compatible with all the aspects which have been reported, more or less, for the multiple sclerosis development, even antibody levels, which are being studied even right now on multiple sclerosis patients in comparison to the non-MS multiple sclerosis patients, show that there is a significantly elevated titer against BMMF, a specific type of BMMF in the multiple sclerosis patients, but this needs to be further elucidated and clarified. Now, this was the one phase, the chronic neurological diseases. Let me just state at this moment that other chronic neurological diseases, namely Alzheimer's disease, Parkinson's disease, amyotrophic lateral sclerosis, if analyzed for antigens in some cells of our BMF particles, of at least one or large open reading frame, the rep antigen, as we call it, it's involved, obviously, in the replication of those agents, is present or is visible, at least in a couple of cells, in those lesions as well. But clearly, an etiologic relationship at this moment cannot be stated, but still, a suspicion exists that it may also play a role in those types of diseases as well. Let me come to the second example, of indirect carcinogenesis, namely particularly colon cancer, which we have studied in very much detail, less so breast and prostate cancer, but the evidence is also there, and also a few other cancers. Well, in all these instances, the BMMF DNA and the expression of the rep antigen, as we call it, they are not present initially in cancer cells or the precursor cells, and so they are devoid of signs of the viral genome infection. But the peripheral cells around these lesions clearly contain them to a large degree. I come to this in a moment. Let's go to the next slide. This became clear after Timo Bund, one of our co-workers, was able to develop monoclonal antibodies against various parts of the this one large open reading frame protein, which obviously is a rep protein, as we call it. It is clearly involved in the replication. And these antibodies permitted really the detection of various epitopes in the same protein, which we analyzed here. What came out there for colon cancer is shown here in the microarray tissue section of colon cancers. You see these ring-form structures. These are the Leverkusen crypts, the glandular cells of the colon, from which the polyps and cancer arise. They are negative for staining. But if you see the surrounding tissues, the lamina propria cells, they're heavily stained by our monoclonal antibodies against REP. So there is a rather intensive expression of REP in those surrounding cells, which we can see right now in about 80% of the colon cancer biopsies which we have analyzed so far. They have a relationship at least to the one primary type of BMMF which we find there, whether the remaining 20% are due to others which do not react with our monoclonal antibodies remains to be elucidated in the meantime. But clearly it's a highly prevalent type of infection. Another interesting aspect is that all these infections, at least in the colon, are closely associated or linked with the presence of macrophages, namely the CD68 macrophages, which obviously contribute to the chronic inflammatory reaction and probably contribute to some degree to the immune reaction against these types of agents as well. As Michelle and her group took advantage of the staining pattern of the interstitial tissue between the colonic crypts, which are stained with this serum, made a microdissection of those materials to analyze whether those microdissected stained materials contain the genome of those agents. The answer is a clear-cut yes. But the interesting other aspect was here that after sequencing these genomes, there was, first of all, there were micro mutations of individual nucleotides among the isolates obtained from the same lesion, which suggested that the oxygen radical act on the single-stranded DNA, which is replicating in those instances as well, very actively, as well as a double-stranded DNA of adjacent cells, which turn later on into tumor cells. There are two other interesting aspects which are shown in the lower left part of this picture, namely that you see these red dots with an arrow, that the RNA transcribed here from, particularly with the large open reading frame, contains polyadenylation signals. This is an important observation because obviously that facilitates the translation of this RNA in vertebrate cells, specifically in bovine cells and in human cells as well. And in addition, the micro modifications also contributed to modifications in the open reading frames of the respective genes, which need to be further evaluated in the future. Well, what about the polyps, which are arising as the first precursor lesions of colon cancer? Well, the picture is shown here. The upper part shows such a typical polyp if excised and analyzed for the expression of the REP protein and also of CD68 macrophages. You can see it on the lower part left, the monoclonal antibodies. To the right, the response of the CD68 macrophages. There's a rather chronic inflammation going on in those polyps for long periods of time. In fact, here it's quite easily visible that the adjacent cells at the periphery of these branches of the polyps are the ones which are the progenitor cells for the cancer, which develop subsequently after much shorter periods of time, and they're exposed much more intensively to the mutagenic activity of oxygen radicals, which indeed are active in the internal part of these types of polyps. So it explains the short latency period of the transduction of a polyp into a malignant tumor probably quite well. Now, this is a model which we developed here for colon cancer. Indeed, you will understand only the first part of it right now, but I'll try to explain the second one. We need two components for an infectious role in humans. namely, first of all, the uptake of these types of vesicular structures in the colon, and they settle down in the interstitial cells of the colon, but you need a second one. And the second one is an interesting one, which we will explain in a moment, namely the uptake of a specific glycan, of a neuraminic acid, the N-glycolyl neuraminic acid, which is a terminal component of lectin receptors of the cell surface. And this terminal glycan component determines the function of the receptor as binding ligand for quite a number of pathogens. Most interestingly, for those who are active in acute viral infections, for noro and rotavirus infections, the main reason for mortality of newborn babies who acquire these types of infection, particularly in countries where the economic situation is very poor. High mortality in India, for instance, for newborn babies is to noro and rotavirus infections mainly. But these types of glycans are also responsible, as I will show in a second, for the binding of our BMMF factors. The human milk contains, interestingly, a number of sugars, 2' and 3' fucosolactose, and a rather complicated molecule, diacyllyl tetraose, which binds with high avidity to the very same receptors and blocks them, if they are present, for the binding of those types of agents, also of our BMMF agents. And this explains why babies which are breastfed for a long period of time are protected against noro and rotavirus infections, remarkably, and also to BMF infections. Indeed, there's also protection against diabetes mellitus, which is something quite interesting for us and which we analyzed in more detail. Now, interestingly, we find the same pattern in breast cancer here. It's not so pronounced as in colon cancer, in breast cancer, but what you see here is a breast cancer biopsy where the glandular tissue of the breast is completely free of the staining for these types of infectious agents, but the adjacent tissue on the upper left part, upper left and right part of this picture are clearly stained by monoclonal antibodies, specific monoclonal antibodies, the same ones which stain the colon tissue quite well. And CD68, again, the macrophages are present here in large numbers and in high concentration in the same type of tissue. This shows a picture for prostate cancer. Again, the glandular tissue is free of any kind of staining, but the adjacent tissue contains, again, CD68 plus the respective staining for our antigens. So we haven't had the opportunity yet to get isolates very well from those types of tissues, breast and colon and prostate cancer, but the evidence right now points to the presence of those agents as well in those lesions, but it needs to be further studied in the future. We are not entirely sure whether this represents the same types of which we find in colon cancer or different types, but that needs to be clarified. So most of these cancers occur late in life. It's very likely that long latency periods exist for all of them, decade-long latency periods, pointing to an interaction of several factors in their pathogenesis. Chronic infection linked to cancers usually require and uniformly require additional events, genetic or epigenetic modification in host cell DNA prior to tumor development, and the frequent interaction with other kinds of mutagens, be it chemical, physical, or other biological factors, have recently been described. We worked on this for quite a while. Every type of mutagen will act synergistically in these types of cancers, as for instance smoking does in colon cancer as well. Now, my wife gets already nervous that I talk too long. 3 phases of bovine factors right now again. The third one is diabetes mellitus, and I will conclude more or less with that one. BMF infections are prevalent in insulin-producing beta cells of the pancreas, of the Langerhans island, and this leads to insulin reduction, to increasing beta cell depletion, and dependence on insulin injections in the late phase of the disease. And there's also, interestingly, a BMF persistence in adjacent macrophages, different type of macrophages, not CD68 macrophages in the pancreas, but macrophages. This results in large aggregates of BMF rep proteins in those cells, in the beta cells, possibly also aggregating, or that's the hypothesis at the moment still, this amylin, which is frequently present in these types of cells. And both of them show in the electron microscope a prion-like state of these types of proteins which aggregate in these types of ways. Okay, thank you. I have 5 minutes. I will try my best to do it. The BMF present in macrophages, chronic inflammation, oxygen radicals, mutations after long cancer periods, increased risk. That has been shown quite reproducibly, increased risk of diabetes type 2, of pancreatic cancer in diabetes type 2 patients and vice versa. Increased risk of also of diabetes type 2 and in cancer patients, and that's the opposite as well, of in cancer patients also of diabetes type 2 and under those circumstances. There's one important factor which I cannot let out here, namely we have in humans a very specific situation. I talked about the glycans which determine the binding of receptors, and that is here demonstrated in this picture. Usually in most animals there's a glycan, an N-acetylneuramidic acid, we call it NU5AC, is converted by the hydrolase to the N-glycollyl neuramidic acid, which is a binding factor for rota and norovirus and those which I discussed before. In humans, this type of enzyme, which for simplicity I call CMHAH, is partially deleted because there's a deletion in exon, which by the way also exists in chicken, almost the same deletion, and chicken meat is always considered as very healthy in comparison to the red meat, the bovine meat. Now, the humans are unable to synthesize NU5GC, but we take it easily up by dietary consumption. And whereas in most animal products, dairy products and meat, they are abundant in relatively high concentrations, this NU5GC. And this sialic acid is taken up by the diet and becomes incorporated commonly as a terminal component into the membrane glycoproteins, which I mentioned a moment before. Now, what is in diabetes? If you look into in pancreatic tissue, there's one interesting question. First of all, do the islet cells from which the insulin is being produced, do they have something which binds our factors or not? And we obtained an antiserum against NU5GC. Humans that do not produce it. Therefore, it's a xenoantigen. We produce antibodies against NU5GC if we take it up, in contrast to the animals which produce it themselves. And so we got from a group in San Diego, Nisivaki specifically here, an antiserum against NU5GC and tried to stain such kind of sections. What you see here is the Langerhans Islands indicated by the arrows here, which are being stained in the cytoplasm, not very strongly, but they're clearly stained in contrast to the remaining tissue. That opens the question, of course, do they bind something like our BMFT agents? And again, with the monoclonal antibodies, by staining here in a low magnification, the Langerhans Islands, you see they're all stained, almost uniformly in those cases. Here of a patient, I think this was a patient with pancreatic cancer. They're all stained, which shows that there seems to be an uptake of those individual cases. And when you look at it after staining a higher magnification with monoclonal antibodies, here for instance on the left-hand side, is clearly a rather remarkable big inclusion bodies visible with our monoclonal antibodies. It's not visible without the monoclonal antibodies. There's a suspicion that this could be from amylin, which has been demonstrated in pancreatic islands, which is a similar type of molecule and which is shown on the lower right-hand part of the picture, but it may aggregate together with the with the staining of the monoclonal antibodies here, which we don't know at this moment, but this is quite a possibility. This shows normal pancreas patients which are due to accidents died, and we see already in the early stage some of this type of staining which increases with age. Subsequently, here again a higher magnification. These are cytoplasmic inclusion bodies under those circumstances, clearly heavily stained by our monoclonal antibodies. And an electron micrograph of these types of pictures, shown in the lower left part of this picture, shows that in the same section of one individual island cell, beta cell in this case, shows something like 5 individual structures shown on the lower right-hand part of this picture, which clearly conform with our BMMF type of particles, which we could demonstrate also in other tissues here, which shows that there must be a very massive infection taking place. And so there's apparently an interesting link between diabetes type 2 and pancreatic cancer. As I said before, there's an inverse relationship. Both increase under the conditions of the one disease, the other one is increasing at the same time. But it seems that there's a common pathway in addition, which leads to the production of BMMF particles, the excretion of the infection in the pancreatic beta cells, and subsequently to chronic pancreatitis, metabolic dysfunction of the pancreas, which has been identified in the past as a precursor lesion from pancreatic cancer in particular, which may lead eventually in pancreatic cancer without CD68 macrophages, but we do find other types of macrophages which are affected here in these cases. In the case of diabetes, the situation is quite similar to colonic cancer here. In this case, mutations due to infected macrophages and the chronic inflammation which takes place, eventually leading to this type of cancer. The incidence of cancers in patients with diabetes type 2 has been repeatedly reported to be for quite a number of cancers there: hepatocellular carcinomas, non-hepatitis A and B carcinomas, pancreatic cancer, endometrial cancer, colorectal cancer, breast cancer, kidney cancer, bladder cancer, gastric cancer, ovarian cancer, and even for non-Hodgkin lymphomas, T-cell lymphomas and leukemias. And again, I show the particles. The interesting question is, since the cattle contain these particles and the binding factors for these particles abundantly, why do they not, as well as other ruminants, not develop cancer of the udder and the colon? The answer is quite simple and probably very straightforward, because all of them are able to produce Nu5GC from Nu5AC, because the enzyme is not defective in those animals, and they're immunotolerant against Nu5GC. No inflammation, no immunoreactivity, no oxygen radicals, no antigenic, no mutagenic activity under those conditions. That makes it difficult to use animal models for these types of studies. This shows the human sugars which are found, which protect against the infection with those agents. Time doesn't permit me to go into the details, but let me just finish by making one brief statement about another leukemic disease, namely acute myelogenous leukemia. There was a remarkable similarity, as shown in the lower part of the picture, between the global incidence of acute myeloid leukemia and also of colon cancer. The regions with high incidence of the one have a high incidence of the other as well, accounting for Europe, Asia, Australia, and also for Argentina as well under those circumstances. The interesting part here is that the epidemiology of those leukemias, the childhood leukemias, include here in both types of childhood leukemias, acute lymphatic leukemia and the myeloid leukemia, that prenatal occurrence occurs. Not very frequently, but even at a remarkable incidence still for both types of leukemias. In the myelogenous leukemia, slightly less than in the acute lymphatic leukemia, and then it goes down. In the acute lymphatic leukemia, it goes up subsequently after delivery to a relatively high peak for a short period of time. Well, right now in mass spec analysis, we could analyze that particular samples from childhood myeloid leukemia do contain the evidence for BMMF infections to a relatively high level. We have multiple hits in those types of samples. For acute lymphatic leukemia, the data are still missing to analyze this. So what we can say, and I'm coming to end my talk, BMF infections are commonly events early in life and lead to slow, probably usually after the weaning period of babies, and lead to long, slow, long-lasting inflammatory reactions. Cancers, as a consequence, appear only after decades later. This represents the most common zoonosis. I mentioned this before. And in colon tissue, the cells of the lamina propria and subsequent inflammatory reactions by oxygen radical synthesis, which we can demonstrate there, play a decisive role for the further progression and the final malignant conversion. Therapeutic interference with these inflammatory events, for instance, by antiphlogistic drugs, aspirin, ibuprofen, COX-2 inhibitors, and others, will have a preventive effect. It has been shown already in quite a number of publications for colon, breast, and prostate cancer. I warn, however, for the uncontrolled intake of those types of drugs because they lead to bleeding types of problems. Preventive approaches need to consider the avoidance of BMF infections from contaminated tissues, contaminated nutrition, and effective neutralization of the factors prior to infection. This is a group who contributed to this type of studies in our cancer center, and in addition, the Swedish collaborators, in particular, Thomas Olsson, Hans-Olof Adami, who is not on this picture, also contributed to the interpretation, and Hans Grönlund were quite active, and a couple of others, which time doesn't permit me to go into the detail. But let me just make a note, a slightly amusing one at the end of this talk. During the past decade, a number of scientists, mainly epidemiologists and journalists, expressed their distrust and doubts about our results quite pronouncedly. Well, arguments among scientists, but clearly also among journalists, are not rare. But it's very rare to catch a photo of those disputes exactly in the right moment. And this is shown in my last slide, this one here. You can see here, kind of judge who is the scientist and who are the ones who are disputing the reality of those results. Anyway, it's just a joke. So thank you very much for your attention.
Thank you very much, Harald Zuhausen. Now please welcome on stage again Eva Kondoroshi, who will introduce our next speaker. Katalin Carico, Senior Vice President of BioNTech SE, Professor at the University of Szeged, Adjunct Professor of Neurosurgery at the Perelman School of Medicine at the University of Pennsylvania, and named Hero of the Year 2021 by Times Magazine.
No. So, ladies and gentlemen, this is my great pleasure to introduce you Katalin Carico. Kati is not a Nobel laureate, or not yet, but perhaps she is the most famous scientist known in this day all over the world. She is a biochemist, born in Hungary, and started her scientific career at the Biological Research Center of the Hungarian Academy of Sciences, where Kati and I worked together. So Kati was at that time a PhD student, and we worked together how to introduce DNA into human cells with the help of liposome. We succeeded and published the paper in 1985, and after Kati moved to the US with her family. She developed the mRNA technology over many, many years without any appreciation or getting any grants. The COVID pandemic had to come to show the enormous value and the power of her discovery. Her invention, in collaboration with Drew Weissman, made possible the development of the mRNA-based COVID vaccines that saved millions of lives. And surely their pioneering work will revolutionize the medical care globally and all the therapies. So Kati, the floor is for you.
Good morning, everybody. I am very happy to be here. Thank you for the organizer inviting me. I am second time in Vienna. First time I was here in 1977. First time when I had the opportunity to see what is behind the Iron Curtain. So I came here as a student. And so I am happy to present to you about the messenger RNA. And I plan to present about the past, how the messenger RNA was developed to therapy, but the main interest right now is to see what is the future holds. So I am talking about mRNA for therapy, but what is the future? Messenger RNA was discovered in 1961. Two consecutive papers describe a very labile molecule, which is coming from copying the information from the chromosome, and this messenger RNA will carry this information to the production of the protein site, which is in the ribosome. So 1961, we know that there is a messenger RNA, and of course, discovering the mRNA didn't make possible to synthesize it. So in the early time, people just isolated RNA. Sometimes, they use viruses, and demonstrated and analyzed what it can be done with it. For me, the most important milestone was 78, which we were reading about these experiments that isolated mRNA was delivered into mammalian cell by liposome. And I learned how to make liposome from Eva Kondoroshi. And in her laboratory, we delivered DNA. At that time, not an isolated RNA, but it was this kind of experiment stuck with me and was very inspiring for all in my life. I didn't know at that time that how revolutionary all of the research was done, which I participated that time. In our field, the next important time was 1984, when Harvard scientists described how to make RNA in a tube. And Douglas Melton and Paul Krieg describe it. And I might mention here that when I contacted with them to get this kind of template that I want to also make RNA, they put in an envelope and they sent it. At that time, there were no transfer agreement, and everything was, every scientist wanted to help each other. So I am very grateful to them also. And many, many scientists, which I emphasize, that who have to develop this field and work very hard. In 1990 was the first time when a paper described that messenger RNA injecting to an animal can be translated to a protein. In 2000, with my colleague Drew Weissman, we discovered that this RNA, which we are making from the four basic nucleotides, actually it is inflammatory. And it was 2005 when we discovered what are those receptors this RNA is activating, and we also discovered that how can we make the RNA non-inflammatory. It was important for me because I was working every time, when I was at the University of Pennsylvania on this cardiology department or the neurosurgery department. I want to make mRNA coding for a therapeutic protein. I always want to make therapeutic protein coding RNA. Meanwhile, my colleague, Drew Weissman, was interested to develop messenger RNA for vaccine. So it is very important, again, that we educate each other. I am the RNA scientist, he's the immunologist, we learn each other. And the next important in timeline is 2017, when first time discovering that this non-inflammatory, non-immunogenic RNA actually is better for vaccine as well. And LNP formulated RNA was delivered into animals and demonstrated that how excellent vaccine these are. Now today we know that the LNP1 component acting as an adjuvant in this vaccine format. And 2021, so 60 years after discovering mRNA, it was the first mRNA product was FDA approved. So today I don't want to describe all of this background, just very shortly, and try to focus on what is in present and what is the future using mRNA for therapy. And one example I will take, how it is used for treating heart disease. One is liver disease, and one is for cancer treatment. So at the beginning of these 1990s, you usually could read one paper per year about RNA. And everybody knew everybody who did something with mRNA, so we were a very close group of scientists. And in the 1990, the first paper which described using messenger RNA, this was vasopressin coding RNA in an animal model as a Brattleboro rat, which has diabetes insipidus, and they used scientists at Scrip, Floyd Bloom and colleagues, and they demonstrated that they can, injecting this vasopressin RNA to the brain of the rat, they can treat it for a couple of weeks. In 93 and 94, two papers where messenger RNA was described for infectious disease vaccine. So that was the first two papers. The first one in French scientists, Maulin and Martinon, they did not use naked RNA as for the vasopressin was used, but they formulated in liposome, just we did with Eva together in Hungary. And scientists in Karolinska, they injected self-amplifying mRNA in a naked form to the animals, and they demonstrated that messenger RNA could be good for vaccine for infectious diseases, influenza. In the following year, in 95 and 96, and subsequently, you could see that most of the publication is about using messenger RNA for cancer vaccine. Curiel and Conry, Alabama, that they used this naked RNA and they injected to the tongue, actually, the mice, and they demonstrated that it can be useful for cancer vaccination. But the most important was in 96, Eli Gilboa at Duke University, who did use the messenger RNA ex vivo, and he established the first RNA company, Merix, out from Duke University, and they initiated human trial. So it was in the 1990s, and in that case, the samples were taken from the patient, and the treatment was ex vivo. And the company later became Argos, and they went a clinical trial all the way to phase 3, but it was not successful. This ex vivo treatment was not helping to the patient. And so 2000 was the next time when an RNA company formed. Actually, it was CureVac in Tübingen, Germany. And their goal was also to use, and they were also inspired by Eli Gilboa, to use messenger RNA for cancer treatment as a cancer vaccine. And the first was for prostate cancer. So these studies are ongoing, and but first we are focusing on the infectious disease vaccine, which was an important milestone, what I consider. In 2017, my colleague at the Norbert Party at University of Pennsylvania, who generated messenger RNA coding for Zika virus specific envelope protein, and this was nucleoside modified mRNA. It was formulated by Aquitas into the lipid nanoparticle and was vaccinated first mice and later monkeys. And why is it important? Because this vaccine was almost identical what is used here for the COVID-19, but this was, of course, against the coding sequence was against the Zika viral protein. and coding the protein. And what was important is that what dose was sufficient for a mice, it was sufficient for a monkey as well. Other previous scientists tried to develop actually DNA vaccines, and what was interesting is that it had to be unproportionally increase the dose for larger animal, in that case. But here it was that showing here that 50 microgram mRNA was sufficient to prevent infection of the monkeys. So this was went on, and Norbert, colleagues, and me included, we did studies and demonstrated that such a vaccine could be used for other diseases. All of these were performed in animal studies and shown for HIV, influenza as well, and herpes simplex virus. And Moderna, the company in Boston, they started using similar formulation with lipid nanoparticle, nucleoside-modified RNA to vaccinate against influenza. And for many people were not aware of that these kind of vaccines already were tested on human when the corona came. And from 2013, I worked at BioNTech, and we already were ongoing collaboration with Pfizer from 2018 to develop influenza vaccine. And when 2020, we switched over, instead of the influenza, to perform and generate a vaccine against the COVID-19. You are very familiar with the scenario that getting the information in January 2020, it took quite short time, but all of the preparation made decades before to make the vaccine in such a time frame. And in 2021, this vaccine was approved. So the people asked that, many of them thought that this was the first time that messenger RNA is used in clinic, and asked the question, so what is the future? But turned out, actually, the future was already in the past, because messenger RNA was in human trial, entered much earlier. And one of them was for VEGF-A mRNA, which was used for treatment heart failure. It started at Harvard University, and this paper was published in Nature Biotechnology in 2013, showing that nucleoside-modified RNA coding VEGF-A mRNA and in naked form injected to the first in the mouse heart, and later in was rat and larger animal. They demonstrated that it accelerates new blood vessel formation in the heart and improves the circulation, improves the capacity of the heart of different animal models which had heart failure. And in 2020 already started certain trials. First, in necrotic wound, they also demonstrated that increased vascularization helps, especially for diabetic patient, necrotic wound to heal. And later it entered to trial for the heart injection. And this was actually the news just in this month, in 2022, that the phase 2 trial where VEGF-A mRNA was injected to the heart of the patient undergoing coronary artery bypass surgery, that not only increased vascularization, but as a result of vascularization, the heart performed better. So this clinical trial will move on to phase 3, and this was for AstraZeneca and Moderna clinical trial. So many of the scientists who work on the mRNA field thought that maybe that will be the first RNA, messenger RNA, that will be approved by FDA, but we know that the pandemic accelerated and pushed the messenger RNA to the front. But again, telling it's important that the messenger RNA, this is nucleoside modified RNA, the similar which was used for vaccine. It is not with lipid nanoparticle, it is a naked form, but already was in human trial, and it proceeded very well in this. The other success we can report, again, nucleoside-modified RNA coding for Cas9 mRNA was used by the company, again, in Boston, is Intellia. And this is a very devastating disease where amyloidosis causing multi-organ failure. And the liver is producing a protein which is very toxic to this patient. So what in Intellia they were doing, that they generated messenger RNA coding for this Cas9 protein. This protein with the guide RNA, which they co-delivered, can have a sequence which is specific for this gene producing this from which they're producing this toxic protein, and they co-delivered in a nanoparticle to the patient, and the liver, they could successfully eliminate production of that. And here it's shown that the messenger RNA coding for the Cas9 editing enzyme translated in the cytoplasm associate with the guide mRNA, which will help to enter to the nuclei and help to edit in the chromosome. And as a result of it, as you can see, this toxic protein is not made. Their clinical trial included treating 6 patients, and 3 of them who get the higher dose could already, they demonstrate permanently were cured. This, might mention, this same disease was targeting for the siRNA for the Alnylam company, where, the siRNA had to be injected more than once. But in the case of, messenger RNA therapy, this patient, those 3 patients who get the higher dose, now is permanently relieved for making this toxic protein. The last one I want to say about cancer treatment. So, as I mentioned, cancer treatment was in the focus of those who worked on messenger RNA, and those mostly were to deliver messenger RNA coding for cancer antigen. So those were vaccines. But what we have done at BioNTech is that we generated a messenger RNA coding for cytokines, that interleukin-2, interferon alpha, GM-CSF, and IL-15. So what happened when this mRNA was injected to a cell surface tumor, here shown in an animal model? So when the cell surface closed tumor, and here, can be used for melanoma in a human setting, and head and neck cancer, where there is a tumor close to the surface, this messenger RNA mixture is injected, and in the tumor, the protein is made locally. And these proteins will invite different immune cells, and they are coming and recognizing the tumor type, and then when they are circulating, that they can perform an abscopal effect, meaning that when they find remotely located tumors in the body, that they can eliminate. In that case, the picture shows that how already established lung cancer was eliminated by such treatment. And this was published last year, but this already is in advance to clinical trial by BioNTech and Sanofi. So this is a human trial again, and it is already ongoing for more than 2 years. And so again, just demonstrating that when people thought that the mRNA coding for the vaccine is the first one, what was used in human, just demonstrating that all of these therapies for treating heart failure, treating cancer, or treating the liver, disease, this already advanced in human trial. So, of course, I mentioned you these several clinical trials where nucleoside-modified mRNA was used for diseases. And in addition, there are also human trials were used where passive immunization was used. So the messenger RNA was coding for antibodies. And the advantage of messenger RNA is very simple to synthesize because it is always made from the basic nucleotides, and in changing the uridine to pseudouridine or 1-methyl-pseudouridine triphosphate during the synthesis is the only one what you have to need to perform. And some of the experimentation, these are used in lipid nanoparticle formulation or in a naked form, or preclinical trials are ongoing for other diseases, and one of them was published a year ago by scientists at BioNTech. What we have done it was for tolerization for multiple sclerosis. In that case, of course, it was the lipid particle did not contain any adjuvant inducing effect. It was tolerizing therapy, and these animal models, two different multiple sclerosis models, the messenger RNA coding for the autoantigen was delivered repeatedly in a non-activating manner and could induce the tolerization in those animals. These are, of course, all of these trials had to be prepared. So these were animal studies. With colleague in Hungary, we also did messenger RNA coding for VEGF-C mRNA, where we could generate new lymphoid vessels, which could be used eventually to treat edema in many different forms. But the mRNA, which is presently in clinical, which is presently in animal trials, is just so long. I cannot list all of them. Of course, now that with the success of the vaccine, many of the many of the treatment is focusing on using vaccines. Those are which we very badly need for treatment and protection against. And so one of them actually last week was announced that Moderna injected the first patient for HIV-specific mRNA for vaccination purposes. So you will see now that weekly that new therapies using messenger RNA is initiated. There is a big community. Those from 2013, we are gathering in mRNA therapy meetings, and those people were aware of that messenger RNA can be used for many different therapeutic purposes or vaccination. You already could hear years ago that scientists already doing animal studies for vaccinating for malaria and many other viral diseases, some or also for bacterial disease. And those are vaccination or therapeutic purposes. There are companies all over the world focusing on mRNA. Again, we were just aware of those who were in the front line or spotlight, might say, during the pandemic, but there are many, many more, and I hope that many, many more scientists will be inspired and will be scientists, try to do, because there is so much work to be done, and so for that I would like to thank your attention. I just show here, some of my colleagues, because, all of this science was done by many of them. Some of them worked with me for several decades at the University of Pennsylvania or at BioNTech, and we work very, very hard and enjoy all of this. And so thank you for your attention.
Thank you very much, Katalin Carico. Next up on stage is Dan Schechtman, Nobel laureate in chemistry and vice chair of the advisory board of the Vienna Congress.
Ladies and gentlemen, we have just seen the power of science. This was a fantastic example of the importance of science that touches each and every one of us. The vaccine that was developed by BioNTech, Pfizer, it was injected to me 4 times already. So we are family now. Can you change the screen so that I can see what yeah, thank you. So this is me, and I'm from the Technion, now retired. Okay. I would like to talk about several aspects that are linked together. I'll talk about demography. I will talk about science, education, and technological entrepreneurships. All these 4 are linked together. And I will start by comparing Israel to Austria. About same size of population, GDP per capita, Israel is 44,000, Austria 48,000, just about the same. But there is a striking difference. Of course, Austria has long history, Israel is a young country, but there is a striking difference in the demography. This is called the demographic pyramid, and this is the demographic pyramid of Israel. And up there, it gives you age groups, and sideways, it gives you the number of boys and girls in each age group. Red is girls, and blue are boys. So, as you see, in Israel, there are very many young people, and this is truly a pyramid. It looks like a pyramid, and it's truly a pyramid. As you reach the age of 100, men are gone, and a few women are still dancing there. Now, this is, oh yeah, and the number of children per woman in Israel on the average is 3.2. Austria is different. It doesn't look like a pyramid, it looks like a mushroom. That means that there are a small number of children. Most of the population is within the working age. So in Austria, the dependency ratio is 50. It means that 50% of the population are working to provide for the rest of the young and the old. In Israel, the dependency ratio is 2/3 and more. That means that less than a quarter of the population has to support the many young people and the old. So this is a difference. And the number of children per woman in Austria is 1.5. 3.2, 1.5. So what does it mean? This is the comparison. When you have 1 child per woman, this is the family. There's grandfather and grandmother, father and mother, and 1 child. And that 1 child will have to provide for his parents and his grandparents eventually. So this is a typical Chinese family, 1 or 1.5 child per woman. If you have 3.2 children per woman, like in my country, a family looks like this. That's my family. So, yeah. So every young man and woman in Austria will have to produce more GDP per capita to sustain the standards of living and to further develop the country. And there are not enough young people. So what can we do about it? First of all, encourage larger families. And many countries do encourage larger family, but different means. I will not go into details, we don't have time for that. But this is done in many countries, and sometimes successful. And number 2, encourage technological entrepreneurship. And this is one of the subjects that I will discuss. So what is technological entrepreneurship? It is an establishment of a new technology venture. It has to do with technology, has to be new, and it's a venture. Some win, some lose. You take a risk when you're an entrepreneur, and you have to know how to do that. So why is technological entrepreneurship important for the well-being of societies, countries, and the world? Let's look at the world today. First, high-tech startups develop relatively fast. They develop fast and hire many more employees relative to their size than large corporations. A new startup can double the number of employees every quarter, every half a year. They can double the number of employees. Large companies, if they hire more people, they will hire 1% more. So startups do very well this way, and they require many more manpower and womanpower. And the return on investment can be very, very large, although there are many failures. In Israel now, there are more than 100 unicorns. Unicorn is a company that is private company, not public, private company, that's worth more than $1 billion. And in Israel, we have more than 100 of them. There are less in Europe than in Israel. So the world today, we have struggling economies, not here, not in Israel, not in many developed countries, but in other developed countries, it's terrible, struggling economies. There are no wars between countries, but terrible local tribal wars. Countries don't fight each other nowadays. They threaten to fight. Ukraine situation, but there are no wars. But tribal wars are terrible. And there are waves of refugees, which you see here in Europe. So what is common to all these countries with tribal wars? There's one thing in common to them. They are poor countries. So for these struggling societies, as well as for developed countries, building sustainable economy requires development of human ingenuity. Human ingenuity is key word. Human ingenuity is the most sustainable natural resource, not oil, not coal, no gas. Human ingenuity will be with us, and it will never be exhausted if we work right. And how can we develop human ingenuity? Well, first of all, basic education to all. Basic to all, not only to the advantaged people of Vienna, also in the remote villages. Of course, this may not be a major problem in Austria, but in many other countries, education to all cannot be obtained. People in the capital have good education in many countries, but in the villages, that's not the case. That has to be corrected. Number two, good engineering and science education. Our world needs more scientists and engineers. The more, the better. More scientists and engineers. And government policy and support. Governments should realize the importance of science. And of course, nowadays, with the fantastic example of the vaccination and the upcoming medications against the virus, I guess governments understand it. So government policy and support. Free market economy. Of course, for everybody here, free market economy is a given. Yes, it is good. Not in every country. I gave a talk in one of the South American countries, and when I said in the parliament, free market economy, they said no. Free market economy is neo-colonialism. Okay, bad news. And above all, no corruption. Corruption kills the economy. Corrupt countries cannot develop their economy. This is what we need to do. But that may not be enough. Also needed is development of entrepreneurial spirit and knowledge. Some countries are doing very well without entrepreneurship, but they cannot last for a long time. We need entrepreneurship, we need innovation, and we need not only science and engineering, we need entrepreneurs that will take the science and engineering and make it into a product. And we need to create an entrepreneurial ecosystem. Governments can do that. My government does a very good job on that. Can we teach technological entrepreneurship? Is it something that can be taught? Or either you are born an entrepreneur or not? Well, yes, we can teach entrepreneurship, and we should teach entrepreneurship. So who opens high-tech startups? These are engineers, biologists, medical doctors, scientists, computer experts. These are the people that open high-tech companies. And so, whom and where should we teach how to open a startup company? Youth in high schools. Start teaching entrepreneurship in high schools. In Israel, we have volunteering programs to teach entrepreneurship in high schools. And of course, in universities, teach the engineers, scientists, medical doctors how to open a new startup. Teach them how to do that. And technicians, these are the people who can make things in their hand. They can open new startups that will produce innovative products. So, technological entrepreneurship, the class at the Technion. I will go very quick on that. 35 years ago, I invented a class at the Technion, my university, called Technological Entrepreneurship. It is designed to teach our students and eventually graduates how to open a startup company. The class was the largest of the Technion ever. I took the largest hall. It had 600 seats. 800 people came to my class. They were standing in the back. They were hanging from the windows. Smashing success. I ran this class for 30 years, and then I retired, but it continues. I did not teach the class. What do I know? But I know the people who can teach the class. These are the key entrepreneurs of the country, and I invited them to talk to my students and tell them how they did it, how they succeeded. How did they find the money? How did they find the partners? How did they sell the first product? How did they do market survey? And so on, and so on, and so forth. Okay, we'll skip that. About 3 years ago, we conducted a research study. What happened to the graduates? There are more than 10,000 graduates to my class. They are all engineers and scientists. What happened to them? Turns out that 25% of them were involved in startups. That's a smashing success for this class. There are cultural considerations. In many countries, there is fear of failure. In many countries, failure is a shame on you, on your family, on your city, especially in the Far East. I say, no, failure is okay, start again. Entrepreneurs fail sometimes. That's not a shame. You are now an experienced entrepreneur. You have a second chance, build something new, and you have a better chance to succeed in your second attempt. And so, this brave new world of startup nation will enable high standard of living, peaceful society, and wonderful opportunities for the educated. Those who are not well educated will lose the battle. So we need to educate the society. And this is job one of every government, in our opinion, is education. Job one, education. And not many countries realize that. In many developed countries, the number of young people who aspire to become scientists and engineers is shrinking. People want to become lawyers and accountants and do services rather than inventing and producing. And so, how can we convince young people that science and engineering is wonderful? How can we convince them? Well, my answer is start teaching science early, very early. I said this sentence on the radio in 2012, and the mayor of Haifa, the city where we live, heard me on the radio, and as soon as the program is over, he calls me from his car. Hey, Danny, he says, did you say start teach early? How early? I said, oh, teach science in kindergartens. Mayor says, will you do it? I say, will you pay? He said, I will pay, not for me, but for the program. And we started a program to teach science in kindergartens. We started with 60 kindergartens, and we created a program, and we taught science in kindergartens. It continues today, although not 60 are not left, but 30 are left. That was not very successful because the teachers, the kindergarten teachers don't have it, although we teach them how to do it. And then I said I had a television program, which was called the national television, which is called To Be a Scientist with Professor Dan. And I will show you pictures from that. This is from the program on television. They created a set for me, like a laboratory, and we invited 3 children. You see only 2 here, but there is one more here there. And it was a very successful program. Parents and grandparents were sitting with their children in the evening and looking at the programs. And I got big hugs from grandmothers in the street. Oh, Professor Shechbal, oh, so wonderful, teaching young people. And early science education, this is one of the results, a very, very impressive result. This is a science kindergarten. It's also in Haifa. And when you enter the laboratory and the kids are not there, you think that you are in a university laboratory. Oh, wonderful chemical laboratory. And then you see that there are 10 kids sitting there with laboratory coats, performing experiments and receive explanations from the teacher. And this teacher, this particular teacher, this stands by me, has a master in chemistry. She can teach high school, but she prefers to teach kindergarten teachers. And after the class, she sits down with them to make sure that everybody understood what was going in the class. I invite people from all over the world to visit this kindergarten and copy it. I said, take 1,000 pictures, copy this kindergarten. It will be great for your country. And several countries did just that. They sent delegations and they built such science kindergartens. I invite everybody from Austria who is interested, contact me. I will take you there for a whole day, talk to everybody, take many pictures, and create it. Thank you very much.
Thank you, Dan Schechtman. Please now welcome Stefan Hell, Nobel laureate in chemistry, who will join us online.
Thank you very much, and a great honor to be part of this panel discussion and to giving a short introduction about my trajectory in science. Well, I'm doing microscopy, and you certainly know that the microscope, especially the light microscope, has been really key to making many discoveries in the life sciences. But you certainly also know that the resolution of a light microscope is fundamentally limited by the wavelengths of light, by a phenomenon called diffraction. And this is why people thought 130 years ago that this won't get better, until the electron microscope was invented, and the electron microscope attained a much higher spatial resolution, basically down to the molecular scale. And this was a big, big change that led to many, many new discoveries. But unfortunately, electron microscopy also has its limitations, which are the simple fact that you can look only at that material. And also, it's difficult to see distributions of biomolecules in cells, very particular. Because usually, if you want to see a distribution of proteins, for example, what you do is you have to highlight them in a specific way. And this can be done very, very effectively by fluorescence labeling. And if you do a fluorescence labeling, meaning that you attach a fluorescent molecule to a biomolecule of interest, then you can see the biomolecule basically indirectly through the fluorescence label, through the fluorescence emission of the label that is attached to the biomolecule of interest. And therefore, light microscopy never lost its key role for the life sciences. It remained a very, very important workhorse for the life sciences, but had a problem that its resolution was fundamentally limited to about 200 nanometers. So that's about a fifth of a thousandth of a millimeter. And therefore, it was clear that if one managed to overcome this barrier and get a higher spatial resolution, break the so-called diffraction barrier, then it would have a substantial impact on the life sciences. And this was actually what I worked on. But this was not very straightforward to do, because it was believed many years, more than a century, that the resolution of any light microscope, also of a fluorescent light microscope, is fundamentally limited. And this was discovered by a man called Ernst Abbe, who lived at the end of the 19th century in Vienna. And this equation basically reflects this diffraction resolution limit. It's basically saying that in order to be separable, features have to be further away than this distance d given by the wavelengths lambda divided by the number in the denominator, and that typically amounted to about 200 nanometer. And this was written on the memorial here that was erected in his honor, and therefore sort of written in stone. And not only it was written in stone, it was also a fact, because at the time I was a student in the 80s in physics at the University of Heidelberg, and I was asked to work on light microscopy. This was a fact. You couldn't get a spatial resolution that was substantially higher than what you can see now. Now, I was kind of bored actually with this subject, because I felt that this is sort of boring subject that is not really interesting. It was the physics of the 19th century, but I wanted to do something cool in my life. I was interested in doing something that really makes a difference. And so I felt, okay, let's break this diffraction barrier. Let's do something fundamental in this field, because that would be really cool. And so I got interested in the problem and thought about whether it would be possible, whether there would be physics that would still allow one to go beyond the diffraction barrier, although no one believed that this would be viable for physical reasons. And so I tried to convince my thesis advisor, I tried to convince other scientists in Heidelberg where I studied. I went to a famous lab, but even there I couldn't convince people. And so I ended up in a remote place in Finland called Turku, because there was a person who said, okay, you can work here more or less with a substantial amount of freedom, and then you can think about whether this overcoming of the diffraction barrier is viable or not. And so about 8 weeks after I got there, I really realized that there is a way of overcoming this diffraction barrier. And you may say now, and I published it as a theoretical proposal, you may say now, okay, the world would have welcomed that idea. It was not a case. It took me altogether about 5, 6, 7 years in order to convince other scientists to give me the lab, to give me the resources in order to overcome this diffraction barrier and to prove that this is indeed viable. And so I was fortunate in the end that I ended up at Max Planck Institute in Göttingen, where I'm now actually, and they said to me, okay, you might not be right, but at least we give you 5 years, so to speak, to check out whether it works or not. And indeed it works, as you can see here. So the standard resolution was fundamentally overcome with a method that I developed, which is called STAT. It was the first method that really proved that there is a viable physics that allows you to get a better and higher spatial resolution than what people thought for 100 years. And as you've heard, this won me a share of the Nobel Prize in 2014. But I knew at that time, of course, that this increase of resolution, which was then by a factor of 10, was not the end. So this method that improved the resolution from 200 nanometers down to 20 nanometers, but I was convinced, okay, you can do it better. And therefore, even after the Nobel Prize, my people and I went on to get a higher spatial resolution. And indeed, we managed to come up with a concept that really gets another factor of 10, and this is shown in here. So here the resolution is basically now down at the molecular level. So it's of the order of a few nanometers. And if you compare it now with the method that won a share of the Nobel Prize, the STAT methodology is yet again better by a factor of 10. So altogether, we are now better by a factor of 100. So imagine that, say, 25 years back, people thought there is no way of getting beyond this diffraction barrier. There's no way of getting a better resolution. Now we are better by a factor of 100 and basically down to the maximum resolution that is possible, which is basically at the size of the, say, molecules that you want to look at. So the limits are not given by the physics anymore. They're basically the size of the molecules that you look at. So not only that, this has also become commercially available. I set up a startup company. We heard about entrepreneurship from then just a few minutes ago. And so now this methodology is available. And our hope is that the fact that you have a higher spatial resolution by a factor of 10 and by a factor of 100 now leads to fundamentally new discoveries in the life sciences, hopefully to getting fundamental insights into diseases and how these diseases can be treated. But as I said, I am a physicist, I'm not a life scientist. Now, the lessons that I learned out of this trajectory is that, of course, science is done by humans. We must never forget that. And of course, humans like to have consensus. Sometimes there is good reasons for consensus, but it doesn't mean that consensus is the same as reality. So very often consensus does not equate reality. And this is something one has to be aware of. Then the second thing I learned is that if you challenge consensus, if you challenge a certain paradigm, accepted knowledge, it's usually not welcome. As I said, it took me many, many years until I could convince other scientists that it would be worthwhile going. And there were many, many situations where I really felt that I would drop out of science. I was unemployed over a stretch of time. I financed my science through licensing patents to private companies and getting research money in return. And there were many days where I felt, why am I doing this? But what kept me going is the belief that in the end, I'm right and nature will be on my side. So I think perseverance is truly very, very important to making breakthroughs. Very often perseverance is as important as ingenuity because all the resistances that come up on the way have to be overcome, and it can only be overcome by perseverance and by believing actually in the idea. And so my final message is that, of course, if there is a breakthrough, and we've heard it actually in both the talks, both by Katalin Koriko and also by Dan Sheckman, if there is a breakthrough, it always leads to economic value. And this is something that we have to tell politicians, because politicians are usually interested in very short-term improvements, but big improvements, big breakthroughs, as a matter of fact, are those that really make fundamental differences. And this is my final message. Thank you very much.
Thank you, Stefan Hell. Now we are ready to begin our first very interesting panel discussion, discussing the world today between scientism and anti-scientific worldview. Please welcome the moderator on stage, Siert Klöting, distinguished professor at Utrecht University and former president of the Academia Europaea. He will discuss with our prior speakers, Stefan Hell, Dan Shechtman, Katalin Koriko und Eva Kondoroschi.
Dan, can you sit one next? Ladies and gentlemen, first of all, a very warm welcome to our panel session. You just were exposed to a warming up by a number of the panelists, including one online, and the others are here sitting on stage. I think you will agree with me that this is really a stellar panel, and of course that the topic today is very timely. There is a sense of urgency here, and the irony is, of course, that the crisis that we have been exposed to made it even more urgent. And in that sense, the world-famous football player from the Netherlands, Cruyff, had a statement. He said, "Every disadvantage has its advantage." And when I listened to Cathy Carrico, it was very clear that without the crisis, we would maybe have to wait for a few more decades till, let's say, the advances that she reported on were made. And it's, of course, bringing us to the question, is it only in crisis situations that basically we feel that sense of urgency? And is it only in crisis situations where we basically can realize these breakthroughs? The topic of today, future, science of the future, of course, that goes hand in hand with the future of science. And of course, we all know that the future is sometimes unpredictable. And that's the same, of course, for the science breakthroughs. And that's also the same for their impact on society and life. What is also clear that the number of these breakthroughs that were reported in the last hours were the result of bottom-up approaches. Nobody told these people to do what they did. They were often going against the mainstream in science. They were often exposed to conservatism of their colleagues, but in one way or the other, they also got the inspiration not to give up, and then has very nicely demonstrated it. But it's basically something that all of you have been going through. Stefan mentioned very much freedom. The freedom of science is, of course, extremely important. And of course, we also know that depends also on the overall conditions in the society. And we also know that there are countries, basically, which threaten the freedom, not only of the citizens, but also by doing so, the freedom of scientists. So my first question goes to Stefan, who exposed us to his journey going to Finland, where, let's say, they let him do the things. He returned then to Germany after he had the first results. What makes Finland and this place so special that you could do things there which you couldn't do, let's say, in the earlier phase? What can we learn from the Finnish experience? Because we know they are very strong in innovation. We know basically they are front runners in investments in science. Stefan, can you tell us a little bit more about, let's say, the specifics of that institute and the Finnish climate for freedom in research?
Yeah, thank you. It's a very good question. So I'm not sure if one can generalize this and say that that's very typical for Finland as a country. What I can say is that very much depends on individuals. So I was lucky to meet people actually who happened to be from Finland, who realized that this person, in this case me, was on the right track. And whereas the majority of people said this is not going to lead anywhere, yeah, they believe that this can lead to something. So I think it's very, very important to find people who give enough freedom in a way that they say, okay, we believe in this person, or this person has the right attitude. I think sometimes you can see only if the attitude is right and if the general idea is right, but you cannot tell more than that. And if you give enough trust and freedom to the people, and of course some resources, because without resources it doesn't work, then of course there's no hope. Yes, what I appreciate about, if you talk about stereotypes, I appreciate about the Finnish culture is that it can be very pragmatic, and And so that's what I experienced back in the 90s. So a lot of pragmatism, that was not the big egos and things like that. So it was just about facts. And I really appreciated that.
So this was a situation, let's say, where bottom-up went hand in hand with a down-to-earth mentality. No snobism, as I understood from you, but simply do the things. The other issue is, of course, that at a certain moment, the availability of scientific infrastructure and in a number of domains, in particular in the exact sciences, this can be quite expensive. So large-scale scientific infrastructure, which is not always available. For that, also, let's say, sometimes you need a little bit of top-down pressure. What do you see as the optimal mix of this bottom-up and top-down, Stefan?
I think both are needed. If there is too much top-down, then you basically stifle creativity among scientists because they are forced to going in a certain direction that is pre-designed, and we see that very often in most countries. This wish that one makes say improvements in a certain direction, and then there's a huge crowd of people going in one direction. I think there is justification for that as well, because some things need to be developed and then you have to steer a number of people in a certain direction. But as I said, and we've just heard actually from Dan Femcotti, of course, there are big breakthroughs, and the breakthroughs usually come from directions that you do not anticipate. And this is why politicians have to make sure that there is enough, so to speak, say protected space and a protected, say, working environment where, say, genuinely new ideas can evolve. And if that doesn't happen, of course, it's very likely that on the left-hand and right-hand side, those ideas will evolve, and people get stuck with the canonical knowledge. So I think there has to be both. Of course, both to some extent steered directions, but enough freedom on the other hand for people to make breakthroughs.
Thank you. The European Research Council has been a game changer in the European research area. Today, we have a budget of €2 billion per year, funding PI-driven research, so PI-centric. It's all about the people. The only criterion is the originality of the idea and the quality of the PI, basically, and it's a success story. It has changed the landscape, and so we are very fortunate here to have Eva Condorossi. She has served as vice president of the ERC and domain coordinator for life sciences. Maybe she can give us her view on how we can export that model also to the different member states in Europe, in particular to the less research-intensive countries. So Eva, floor is yours.
Yeah, I think the ERC system is really the best. You can come with your original ideas and develop your own research group and perform your research with really very nice funding. The only problem that we cannot use all the talent from Europe. Actually, the 2% of the ERC grants goes only to the new member states or to the European research area. So to those countries where you really need a high level of research and when it should start actually. If you have a few grants in these countries, this is sufficient to change the whole scientific landscape and start really new modern science and really have the chance to realize your dreams and your thoughts. So this can bring something absolutely new. So in most countries, also in the EU, certain countries, so the new member state, usually you have grants. And these are all top-down grants you have to work on certain types of topics. So this does not give enough room to develop your own ideas. And therefore, ERC is really good. But it's a question how we can increase the participation of talents from these countries. What we can see that the most talented young people already at the age of 18, 17 are searched by big Western Europe universities, and they go to this country. So there is no brain circulation, but more brain drain. I can ask also maybe Stefan. He is from Romania, from Arad. And what were the conditions and why you left Romania, and how was your situation? Was it the infrastructure or what else was missing where you had to leave the country and do the work in Europe and other countries?
Stefan, the floor is yours.
Yeah, thank you for the question. Actually, I left Arad, Romania, border to Hungary, at a very young age, at the age of 15. So this was still communist time, as you know, and we were part of a minority and definitely wanted to leave the country and settle in West Germany, which was, of course, a liberal democracy. And without that, of course, I think it would have been very hard for me to make a difference, because there was free education in Germany, free education at university, and I could thrive, basically. And so I owe a lot to this liberal democratic system of West Germany. In general, I think that I agree with you. There's a lot of talent in the world. And I think to a large extent, talent is equally distributed. And we have to leverage the talent that is in those countries where science is not, or science, say, infrastructure is not developed. And I think more can be done. And I very much regret that only a small percentage of the ERC grants are given to those countries, like, say, perhaps Hungary, Romania, and so on, because there is a lot of talent, no doubt. You're saying they are attracted to the West, but sure, but some of them will go back, or they will have, say, enough knowledge in order to set up their own interesting science. As I said, there's a lot of talent there. My take is the following. It's very critical to reassess the way the ERC grants, or grants in general, are given. And my view on this subject, my humble view, is that there's a tendency of reviewing bodies, so those that give out the grants and review those grants, there's a tendency of reviewing bodies to give grants in the same direction. I fully agree that ERC is the best system that we have, but still, if you have a reviewing body, say a commission, so to speak, there's a high tendency of perpetuating the same research direction. And I think there should also be schemes that say, we give the money out there and there's a substantial risk that there will be no return, yeah? But at least what the people are doing is highly original and not based on some consensus on a reviewing body of a reviewing panel. This is my greatest worry. And I think in that case also, perhaps even the percentage of scientists from, say, scientifically less developed countries would have better chances.
What we basically see is that there is a lot of talent around, whatever you look at. We see also that brain circulation is crucial in terms of coaching, exposure to other people. And it's clear that we have the tool in Europe, the European Organization for Association for Cooperation in Science and Technology, COST, the D-networking tool in the European research area. And COST is spending 50% of its budget to the researchers from the less research-intensive countries. And we have the record that these people, after being involved in a COST action, in the networking program, that they have sometimes up to 40% success rate for subsequent applications to Horizon, also including ERC. the successful ERC grantees from these countries most times have been involved in a cost action. So there you see, basically, there's a lot of low-hanging fruit, but these people basically need the coaching. These people need basically the exposure to people outside their country, which are common in, let's say, places like ATH, or let's say in my own lab in Utrecht, which are international by definition. So I think there's more to be done here also, but we have also here a few success stories, but we must speed up, that's very clear. And as former Vice President of ERC, I recognize also a little bit of what you say. Sometimes excellent scientists can also be quite conservative and are not open, let's say, for ideas from corners of Europe where you don't expect them from. I have another question to the panel. As mentioned by Cathy, sometimes it takes quite a long time before something is done with the research findings. You mentioned that some of these basic breakthroughs were made a long time ago. Why did the market didn't pick it up faster? What was the lag? I mean, Dan has mentioned the importance of entrepreneurship, but let's say we know also from certain fields of science, including my own field, which is earth sciences, where basically all the ingredients for exploring successfully for geothermal energy were there already decades ago, but it was not interesting enough for the very big investors, and it was too big for the small ones. What is the situation in your domain, reflecting basically on what you presented here just a few minutes ago?
Yeah, so in these days, actually, more paper is publishing about why, was not recognized the importance of RNA and me, and then try to find the answers that some of them we heard here, like those people who are evaluating the grants, they have too many grants to evaluate, and they look at there and what's similar what they are doing is easier to tell them that these topics should be evaluated. Most of the time I was the bottom, and then they didn't even give reason why were rejected. But so that's one reason. And, I was listening with great interest here, the panel members and the presentation by Stefan. And then, so that the one was not mentioned is this human factor of belief, that the people start to not believe in themselves. And I might mention that when my daughter, she was in second grade in the US, and she was like 7 years old and came home one day, and she said that, Mom, I learned school that I am the only one in the class, cannot be a president of the United States because I was born in Hungary. And then in the break when they had, the others comforted her, for this kind of thing, because all of the other kids thought that they can be. So that's what I think it is so great that some people, they believe in themselves. And so I can see also that people start to settle for less. They lower the bar because they think they cannot do something. So that's why I try to, encourage them to believe in themselves. And so that the perseverance is another thing which, I was quoted many times that, why I could keep going. And what I found is some other scientists saying that they left because, the others were advancing more and they work hard. And so for me, this Hans Scheyer book in the high school, reading them, focusing what I can do and not to focus on what I should have done, not, because already done, and don't waste your time, because you cannot change that other people may be promoted, and and don't work for the boss to please anybody. You have to be pleased to understand what you are doing, and so that you don't get disappointed. So those many things I was thinking that what I learned from this book, and how I could focus on something. Of course, it is important that I focus, and I could see the advancement and the criticism, that I listen. So it is very important, because in our field in science, the critics is making you sharper to, maybe he is right. And, then you do more experiment and you are open for other possibilities. And so not to take personally that somebody's criticizing you, it is about the science. And so many things, the other thing you said that in Finland you found some supporters. So for me also, it might not get the grant, but at least one person, was enthusiastic what I am doing. That was enough. That because we could help each other and move on, and somebody always had money and could do those, very limited amount of what had to be not much, not expensive experiment, not very expensive equipment needed, but we had enough, to support. And so I try to find the answer how somebody could succeed. I have no special skills, I'm not super memory, nothing. So that if you just focus on something, find pleasure, you will be good at, enthusiastic, have some colleague who is supporting you, and then you can do great thing in science and enjoy it.
Yeah, I think that's a very important aspect what you mentioned, the fun. That's very important. You're all role models, let's say, for high risk, high gain research. And my question was also not only, let's say, the barriers you run in, let's say, on the academic side, but also, let's say, sometimes the slow take up by those who can do something with the research, the companies. The people basically who have to bring it to the market. Many of you, and I look in particular to my neighbors here on both sides, have experience in working, let's say, with companies. And myself, I've been promoting 80 PhD students. Half of them went into the energy industry. Conservatism is not only, let's say, there in academic circles, but also in companies. If you compare it, basically, what is your view? Also developments in time. I know some of the big companies in the past had big research departments, but due to shareholder values, the cuts were made first there. What's the situation today in the parts of the industry that you know so well?
Yep. So I myself, with my colleague Drew Weissman, actually, we established the company 2006, RNARX. Nobody heard about. We didn't get our own patent to our company, so it couldn't move. But I moved to BioNTech and Germany, and I found after decades of working in academic that it was so delightful. You didn't have to get any committee to make your CV thicker. You had to make a product which is helping somebody, which is functional. You cannot fake it, you cannot make it longer describing something. It had to be functional. And what was beautiful also for me is that everybody worked together. There were not that you will be first or last author or someone. It was everybody working equally together. We need the formulation, we need the animal model. And then it was, for me, I was very delighted that finally, everybody is for one goal, to make something which is helping somebody.
Thank you. Same question to them. Israel, a place, let's say, where the lines, lines are very straight, often between those who develop the ideas of frontier science and those that do something with it. Maybe it would be good to hear from you, basically, also a little bit more about what Kathy just has been mentioning for the domain of life sciences, also from the domain which is closer to your field of expertise.
I cannot hear you because of the echo here. So I need your help. Can you come here? Yes. Come to me. I cannot hear you over the because of the echo. Can you talk to me? Not to the microphone. Talk to me.
Yes.
Come here.
Okay, I'm here.
Not to the microphone. Tell me what is the question.
So after Katalin Kariko told about her experience at BioNTech and the difference between BioNTech and being in a corporation and academia. Siad Klöting would like to hear your opinion about how it is in Israel, and because he sees that
what is in Israel?
That the lines between the people who develop products
Yes.
and make them happen are very short in Israel.
Yes.
So what is your experience about that, especially when it comes to life sciences?
Okay.
And exact sciences.
This has to do with national character. Israelis are not very polite, but they're very fast in adopting new technologies and are very fast in implementing them into new products. There are several sources to this. First of all, good science and engineering education in several universities, starting with my university, the Technion, but also in other universities. This is one factor. Factor number two is the military service. Today's military service, many people are not fighters. They develop new high-tech technology for listening, for espionage from afar, and they develop an infrastructure of high-tech for the military. Once they are released from the military service, they have all this knowledge that can apply immediately to civilian products. Another factor is the following. Because of the very good manpower, woman power, of the country, many large international companies, any name that you can mention, from Microsoft through Google, through any name, have development center in Israel. There are more than 300 such development centers in Israel, and they employ tens of thousands of very capable minds. Is it good? Well, you can say that these people get very good salaries, so it's nice. But they take manpower from the startups. If a startup is looking for manpower, it's difficult to find and they have to pay a lot of money. However, the bright side of this phenomenon is that these people that work for those development centers know what's going to happen in the world of technology 5 years from now. And if they leave the company, then they can open a startup that will cater to something that will happen 5 years from now, and nobody else knows that this is going to happen. So it has to do with education, very good education, science and engineering. Has to do with national character. People are fast and people are not very polite. People are very personal. Everybody is a friend. Okay, you don't call somebody Mr. So-and-so, you call him David. David, what's going on? And there are no ranks in the society. Everybody is sort of equal. You can talk to your commander or to the chief of the company in person. One more thing. In Israel, you can talk to the CEO of the company directly if you are a bench engineer, no problem. I talk to many CEOs and owners of large corporations in Israel, and I ask them, is it true that a bench scientist or a bench engineer can call your secretary for an interview with you and you will accept him or her to talk? He says, why call my secretary? They can call me. My cell phone number is available to every employee. They want to talk to me, they will talk to me, and I will say, yes, come Wednesday, 10 o'clock, let's talk, let's hear your ideas. And last but not least, CEOs in Israel or managers in Israel initiate group discussions with the lowest level. They will go into a branch of the company and say to the engineers, we want to hear your ideas and your complaints. Let's talk. And they do it on a regular basis. No rank differences. Everybody can talk to everybody. All this ecosystem or technological ecosystem creates very fast response to new challenges and to new ideas.
In other words, a class society is basically death sentence to progress in science and the take-up of science. Of course, we also realize that many of the challenges we are facing are of interdisciplinary nature. And basically, we see that in the science itself, but also in what will be done with the science, not only in bringing the results to the market, but also bringing it onto the radar of the politicians for legislation. After all, a lot of the issues like the energy transition, steps are made there based partly on insights that have come from the science itself. And we also know, of course, that in many situations, in particular in crisis situations, decisions have to be made on the basis of incomplete knowledge. And we have to live with those uncertainties. But I think this is an important topic also for Europe today. We are fortunate that we have a mechanism for this, the Science Advice Mechanism to the European Commission, where the European Commission wants to know where the science stands on issues which are crucial, for example, the Green Deal, but also for cancer screening. And we are very fortunate to have here Eva Condorossi, who is one of the 7 chief scientific advisors to the European Commission. So Eva, what is your view on this? Also in living with uncertainties in the science and responding to, with a sense of urgency to questions that come up from the point of view of legislation, either from the Parliament, the European Parliament, or from the Commission.
I think the politicians should be very well informed, and they should know what is the scientific evidence to certain questions, what are the things which are not known, which cannot be even predicted. And they have to also be prepared that maybe the scientific knowledge will increase and they have to change the legislation according to that one. But they have to know exactly what we know, what is sure, what we don't know, and how to use it as an advice from our side for their decisions and making legislations. I would like to come back to one point, because you mentioned that we need scientific solutions for certain questions, and this should be taken up by companies. We are talking about the antimicrobial resistance since many, many years. And we know that many people die from that one, and there will be many more people who will die because the golden era of the antibiotics is over. But I do not see in Europe or anywhere in the world really the enthusiasm of companies for starting clinical trials and testing new drugs. There are very, very few. And in fact, these type of drugs exist, but simply when I call a pharmaceutical company, they are telling that we are not interested in antibiotics. You can treat the people fast with that one, and this will not bring enough money for them. So there are things what could be done much, much better and link better the need based on scientific findings, and also due to the need for therapeutic care, really to implement that one in the world.
I think that's a very important point. Kathy, what can we do on this?
So actually, for vaccine was not something that was, considered for many companies. My CEO said that in 2015 that, Kathy, that we are a company, and it is a moral obligation for us also focus on infectious disease, because this is, not something that company could make money at. And that's how we started. And I have to mention that in the United States, they also realized that, they are giving money for grants, and then somehow, scientists develop some kind of ideas, and that was that was the end of it. So 2-3 years ago, or maybe 4, the United States gave out the grant, but it included the phase 1. So the scientists had to think about what will be the clinical trial which they had to implement. So that would be also a solution, that all of the academic ideas would, ended up in some product. And so how to encourage people or companies to focus on something which is not profitable, and the government had to step in probably to help.
Yeah, we have seen the development of something in this spirit in the last few years in the Horizon Europe program. I have been very active in this, in introducing in the ERC the proof of concept scheme, which is an add-on grant where people who have received a major grant from the European Research Council can apply to for exploring innovation potential of their findings. And the same thing we have included in the cost scheme, the networking tool of the European research area, also there an innovator grant, an add-on grant to people who have carried out a successful network of researchers. So I think this is very important, this first step. If you have overcome that, then basically you speak the same language as the potential in investors. But there's clearly a need to work on this because Europe is not that good in this. And that's also realized by the European Commission, by the foundation of what is called the European Innovation Council, which is a big instrument, but that's basically already more for step 2 and 3. What is, I think, also important is this issue that sometimes the request for advice by the politicians also reveals that, well, we have major gaps in our knowledge. This is also the case in the topic of microplastics and the biogradation. There we found that the scientific base basically for advice in some domains of this field is rather thin. So although this is an instrument for science for policy, it also had an impact on the policy for science. As Dan has mentioned, education is key. And it was wonderful to see him in action with these small kids and to see, let's say, laying the foundation for a scientific-minded new generation. And we all know how important the role of the teachers is there. And we also know that in the past, many of the teachers had a PhD. And this is not anymore the case. At the same time, we see also the importance of lifelong learning, because what we learned in school and in the early phase is not always adequate for the type of things we are exposed to later. So I have a question here to the panel, how they see the development in the domain of lifelong learning. And I start on the left-hand side from my direction with Eva.
Yeah, I think as a scientist, we are doing the lifelong learning all the time. But I am very much disappointed in certain countries, let's say in my country, Hungary, where the science education went down very much. There are very few lessons, so even biology is taught for 2 years only in the secondary school. So this is not enough time to raise enthusiasm and interest in the young people to continue towards science. So it should come back really. There is another problem that nowadays to be a teacher is not a challenging task in certain countries. Let's say in my country, the salaries are very low, they cannot live from this salary. So actually the old generation of teachers will disappear soon, and there are no new teachers, or very few. Let's say it can happen that in 1 year in whole Hungary, maybe 10 people wants to be a mathematics teacher. So this is not the future. So we have to raise really the education of the society at a much higher level. Otherwise, the country will collapse or there will be some really big problems. So this is a key thing, really, to educate the people and have a much higher scientific knowledge and standard in these countries.
Thank you, Eva. Same question to Kathy. Lifelong learning.
So I think that all of the kids are curious. You know, they want to know. I remember when we had a chicken, you know, and we watched how it's coming from the eggs. And then if people around, whether it is teachers or the parents, you know, that encouraging or saying something, and early on, it has to start. And I don't know right now, you know, in the United States, how the teaching of science is proceeding and how it can, what can be done to encourage the next generation of scientists and what should be done. I don't really try to show up here expert of everything. I just a biochemist and not knowing about that. And just looking at my own example, that in the elementary school, we just did a crystal, you know, and I know that even I am in the US and then I see a crystal in the bottom of the bottle, I look at there to look at the shape. So early on, what Dan was emphasizing, you know, that to have the child curiosity to keep on forever. You know, that's, but I don't know how we can do, but definitely teachers, we need them very much.
And of course, we are forever young. But what about to bring also the older people back, let's say, to science? In my field, in the energy sector, the companies send people often to short courses given by academics. And there, also in our field, we have many extraordinary professors from the industry, which is very, very useful. Isn't that something there where we can do a little bit more?
Yeah, older people or just average person that, you know, yeah, like visiting on a farm or something that the children can see things. I don't know, but I think every child is curious. I remember looking at the store coming back in the spring and we learned that it went so far away, Africa, and then thinking that, how did they find our house? Or, you know, all of the kids is one. And then if you have them to further that, you know, how they orient, you know, or as we get older, we learn more and more, and then we can learn and understand how they do it in molecular level, let's say, when you are 20-something. But so I think that everybody is curious, but people want to know that how the vaccine works. They want to know that what the disease is doing to my body, and the people are curious. Yeah. I think, but we just have to help them to better understand. And during this pandemic, I also realized as a scientist, we have not done enough to educate the public in a simple term, so they don't know how the immune system works.
Yeah, yeah. Stefan, I see you nodding. What are your views on this?
Yeah, I agree with everything that has been said. So, first of all, I fully agree with Eva that and also with what Kotti has just said. Teachers are key and are really key. I mean, without my teachers back in Romania, I wouldn't sit here, to be honest with you. Yeah. Because when I came to Germany at the age of 15, I already knew that I wanted to become a scientist, because I had excellent education in physics, in mathematics, in chemistry back there. And so I got fascinated with it. And just as Kotti just mentioned, I was fascinated as a child to watch nature, to see certain things, and I discovered them for myself. Of course, I didn't discover them, but I discovered it for myself. And this fascination was something that grew in me, and teachers managed to kind of nurture that. And so I kept going. I felt that life as a scientist would be very rewarding. And I think this is something that needs to be nurtured also in our kids, or the young generation. Sometimes they feel that it's perhaps better to be, I don't know what, an actor or something of that kind. I think it's very important that they see role models that are, say, scientists. So they see that they can make a difference with their lives by becoming scientists, by making discoveries, and also be economically successful. I mean, so this is also a part of the equation, that if you discover something, as I said, you can also be economically successful. It's not the goal, but it can happen. Yeah. And I think this is something that has to be nurtured. So we have 4 kids, and they're all right now still in school age, and I watch it myself, and I must say that of course kids are very different, as we all know. Some want to discover something and have this drive, some maybe are not, but we have to pick out those that really want to make a difference and nurture that.
Thanks. We are coming nearly to the end of our panel, but before closing, of course I notice, and many of you notice, that on the podium here, in the panel, also online, we are sitting here as a group of people from life sciences and physical sciences. And we also know that, let's say, it's very crucial to bridge to the social sciences and humanities, in particular, let's say, for the social acceptance of some of the findings made on our side. What can we do better here? Because this lack of sometimes of interfaces hampers, in many cases, also the switch to novel technologies and basically the uptake by the people also of the need to basically take benefit from findings from research, including vaccines. So I start with Katie here.
I mentioned that the educating of the public, educating all of the other people would be very important, and I realized during this pandemic that when people ask me that, microscope, I am watching the RNA, and this was a, reporter would not know that you cannot see under the microscope an RNA. So the lack of knowledge was there, and those who, like reporters and others, so the social communication was not there. They don't know, and as I mentioned, Shaya said, you focus what you can do, so that's what I focused. I try to educate, try to find simple terms and explain complex processes to the public. And of course, inspire the women, the children, because, representing in US also the immigrants, that so people can associate with me and then maybe inspire them and not to give up and be scientists and so that's what is.
But don't you feel that we need to involve more, let's say, people from the domain of social sciences and humanities in these efforts? In my field, also in environmental research, we see basically that sometimes there are barriers that we can probably overcome if we see more efforts in the domain of environmental social sciences. These are profis, and often what we do from our side is doing our best. But sometimes I have the feeling that a little bit of help from people who know more about psychology and about economics and about basically talk in a language which is also, let's say, digestible, not only, let's say, to the general public, but also to the policymakers. And you mentioned the role of the journalists here. Don't you think that basically, also in view of your major efforts, that we need also to find our counterparts in that domain?
Yeah, I mean, I gave an interview. I told the reporters that we are responsible, that, we did not educate the public, and they are also responsible. So together, but I am not expert on it. No. Maybe
we are partners in crime. That's very clear. I move to Stefan. What are your ideas about that? Because when we talk about interdisciplinarity, we cannot exclude social sciences and humanities.
Yeah, actually, I see a tendency, at least within the domain that I'm to some extent overseeing in the Max Planck Society, that even social sciences and even history is becoming more leaning towards the natural sciences, because I mean, people have realized in these fields that if you want to make a big difference, you have to be fact-based and have to find evidences. And so I see these areas also becoming, say, more susceptible to natural science ideas. So I don't see this disparity in the future where there is social sciences on the left and there is natural sciences on the right. I think they will kind of merge. It'll take some time. It won't happen tomorrow. But if in the perspective of about 10, 15 years, you will see that happening. But that doesn't change the fact that we need to explain to the people why science is so critical. But I think the pandemic helps. In that sense, I'm coming back to what Cruyff has said as a football player. The positive side of the pandemic is that it makes it so obvious to everyone that science is the solution to many, many problems.
Thanks. Eva is working in the group of chief scientific advisors, also with social scientists. And maybe she can comment on this point a little bit.
Yes, when I joined this group, we started with a statement on COVID. And then it was continued with a scientific opinion, how to manage pandemics, usually in Europe. And this was done together with a group of ethics and many, many social scientists. And first, I did not understand, especially when we were at the statement of COVID, what they can do, how they can help actually to come out from this COVID situation. And when one of them started that, let's see the gender balance and whatsoever, I was surprised, how on earth gender balance will come here? But finally, they were right, because they were also looking the social conditions, living standards, who takes care of the others, and they are mostly the women, so they were more exposed to infections and this kind of questions. But as life scientist, at the first things, I would not think about it. So they are bringing in many new aspects and ideas what we are not considering, and they are very important finally for the solutions. So I think if we want to solve problems and this affects the whole society, we have to work together with them, and they can provide also very useful opinion and advice how to treat certain problems. So we should not be so we have to deal with complex problem with a multidisciplinary way.
Thank you very much, Eva. We have 1 more minute to go. So then, any final statements from your side? You have the final word. Tell him he has the floor before we close.
Come here.
Give him the final word.
Yes. Would you make some final words?
Okay. Some observations about current situation in the world. Number one, never in the history of humankind did one person affect so many people around the world. This Chinese guy, who was the first to be sick by the pandemic, by the would-be pandemic, is one single person that affected billions of people. Never in the history of humankind did such thing happen. Number two, second law of thermodynamics is true again. It is very difficult to correct, and it's very easy to destroy. And one person could affect so many people, and it takes so many people and so many minds and so much money and so many resources to fix it. Entropy tells you that globally the world is disintegrating. The disorder is growing. So what do? We try to reduce the entropy of the world. We, the scientific community, are trying successfully to reduce the entropy of the world. We create order. We create life. So my definition of a good person is a person that reduces the entropy of the world. What is a bad person? A bad person increases the entropy of the world. It is very simple. It gives you a tool to judge. Right? If you start a war and you kill and you destroy, then you are a bad person. You increase the entropy of the world. If you are peaceful, you educate, you create solutions, you create scientific solutions, then you're a good person. You reduce the entropy of the world. Last but not least, education, I'll repeat what I said before, is job one. We, the scientists, have to prepare the next generation of scientists, starting with our families. Stephen Hell knows one of my children very well. And this child, when he was young, I was used to talk science to him. And one day when he was 5 or 6, he says to me, "Daddy, this cannot be." I hugged him. Wow. He was wrong, of course. But he had his own opinion. I said, "Okay, let me tell you something that you don't know, and you'll see that it can be." I explained to him one of the laws of thermodynamics to a child. He said, "Yes, that cannot be." That child is now a scientist and a professor at the Technion, and he works on about the same subject as Stephen Hell here. So education starts at home. Education means that you should have the best people teaching. The teachers should be the best people. This is not the case, definitely not the case in my country. In my country, the teachers are not the best people. And that's bad, bad, bad. And this should be corrected. I envy the countries in which the teachers are excellent, are role models. So that should be corrected and work. Government should work on it all the time. Better education, basic education, mathematics, physics, chemistry, thermodynamics. Young children are wonderful. You can teach young children science. They will understand it if you explain to them properly. And I encourage you all in your countries, try to do that. Try to bring about science education to young children. If this is what I can contribute to this meeting, wow, I'm happy.
Thanks. It looks like that we have a plan. It looks like that we are on the way. I think you will agree with me that we had a pretty lively panel session, and I would like to thank the audience, people here on site, but also the people online. And of course, I would like to thank very much the panelists. So please give them a hand. Okay, now we have a break and we reconvene in 20 minutes. So,
Yes, that's correct. Please
tada.
be back at 1:20 so we can start on time. Thank you very much to the panelists. Glad you all came back, and I'm sure you won't regret it. Because now we are coming to the next highlight of today's Congress, the Golden Arrow 2022 Lifetime Achievement Award ceremony. To get us into the right mood, let's watch a short movie. The Lifetime Achievement Award Golden Arrow is annually granted to people for realizing extraordinary, visionary ideas in an exceptional manner, for making the impossible possible. To start the ceremony, please welcome on stage David Unger-Klein, the initiator of the Vienna Congress com.sult.
The Lifetime Achievement Award Golden Arrow of the Vienna Congress 2022 for her life works goes to Katalin Carico. Through her decades of research on RNA-mediated immune activation, she has made a decisive contribution to mRNA vaccines against COVID-19, giving mankind an effective tool to combat the global pandemic. Her research work on the therapeutic use of mRNA opens up completely new medical possibilities for combating other diseases. Katalin Carico wrote research history that is already protecting hundreds of millions of people from a serious illness and opening a new chapter in the history of medicine and health. The Golden Arrow of the Vienna Congress 2022, the Golden Arrow 2022, honors a research career that is second to none, and it's an impressive role model for new excellence in the field of medically relevant research. Congratulations.
Thank you, David Unger-Klein. Now I'm delighted to welcome again Dan Schechtman, Nobel Laureate in Chemistry and Vice Chair of the Advisory Board of the Vienna Congress, to give his first laudatory.
Ladies and gentlemen, Katalin Carico is an impressive example of the spirit of research that has been dedicated to a topic for decades. She has made breakthroughs after many setbacks and has gone the way for basic research to implementation in the form of vaccine product affecting each and every one of us. Only a few succeeding in the way she took. It is all the more important to put the spotlight on these, and we need to make more success stories like this possible through an innovative system that promotes the spirit of research and entrepreneurship. Through the communication of role models like Catherine Noriko. Role models for young people are so important. We have to bring those role models to the young people. The golden arrow has so far gone for a few older men, but now it is going to, quite rightly, to comparatively young, very wise women. Congratulations.
Thank you very much, Dan Schechtmann. The next laudatory speech will be given by Thomas Seckeres, President of the Austrian Medical Association and the Medical Association of Vienna.
Distinguished Professor, dear ladies and gentlemen, I'm particularly pleased to be the laudator today when Professor Kariko receives the Golden Arrow of the Vienna Congress for her lifetime achievement. She will share this award with some other great personalities such as Shimon Peres, Lech Walesa, Hans-Dietrich Genscher, or Mohamed Al-Baradei. Katalin Kariko has changed people's lives, not through political initiatives, but through scientific excellence. The fact that there's an effective instrument against corona is mainly due to her research and her achievements. The focus on her biochemical research is on RNA-mediated immune activation. Together with the American immunologist Drew Weissman, she has investigated the possibility of suppressing the immunogenicity of RNA by nucleoside modification. This technology enables the therapeutic use of mRNA and formed the basis for the development of mRNA-based COVID-19 vaccines. Professor Kariko studied at the University of Szeged, not too far from here in Hungary, where she also received her doctorate. Already during her time in Szeged, she worked on the synthesis of RNA. After her immigration to the USA in 1985, she continues to intensively research this field. And together with Drew Weissman, with whom she researched the development of mRNA-based medication. Unfortunately, it was the university that sold the patent on the medication and not her or her partner. Nevertheless, she remained true to her field of research. She finally succeeded in modifying the RNA molecules in such a way that they are no longer attacked by the immune system, and these results were published together with Weissman. Derek Rossi of Harvard University took up the technology and further developed it. In 2010, he founded a company named Moderna with colleagues. And we do know this company at another company which is producing an effective vaccine against COVID-19. In Germany, the founder of BioNTech, Professor Sahin and Turezi, became aware of Kariko and offered her a position. She has been senior vice president at BioNTech since 2013. The aftermath, ladies and gentlemen, is history of research and medicine. It's an open secret that such a scientific life doesn't remain without awards. I've stopped counting at around 40 awards for Professor Kariko's achievement so far, but today she receives a very special award, the Golden Arrow of the Vienna Congress for her lifetime achievement, which is particularly due to her, for a lifetime achievement that secures and protects all of our lives. Thank you.
Thank you very much, Thomas Sekeres. And now it's a pleasure to welcome on stage Elisabeth Köstinger, Federal Minister for Agriculture, Regions and Tourism of the Republic of Austria.
Dear Professor, distinguished guests, thank you for having me today. On Twitter, Delta Airlines recently asked a seemingly innocent question. Name a city that changed your life. And a jokester replied, Wuhan. I think we all agree that the past 2 years have challenged all of our lives. As mothers, as fathers, as husbands, wives, work from home employees, but most importantly, as human beings. Turns out the world is even smaller than we ever thought. Every crisis bears an opportunity. As French poet Victor Hugo once said, there is nothing stronger than an idea whose time has come. Professor Carico, your time has come, finally. After more than 4 decades of believing in your idea and research, you're getting in the spotlight that you and your groundbreaking work really deserve. 2 years ago, only very few of us knew the name of Wuhan. Even fewer knew about the work on RNA and how genetic information can be converted into proteins and vaccines. Your revolutionary discoveries will continue to impact billions of peoples, and we all here stand small before you and your life dedicated to science. You are quoted as saying, I never had any doubts that it would work. But your career was not free of setbacks or disappointments, and your work was overlooked more than once. But you pursued your research with the utmost discipline and rigor, even against yourself. At some point you slept in your lab because you were so consumed by your work. There are few people who dream about changing the world for the better. And even fewer people live to see their dream become reality. The Golden Arrow you receive today is more than just an award. It's a symbol of perseverance and courage. Professor Carico, you are a role model for all of us. I beg you to continue to believe in your dreams. Thank you.
Thank you very much, Elisabeth Köstinger. And now please welcome on stage our next laudatory speaker, Katharina Reich, Chief Medical Officer at the Federal Ministry of Social Affairs, Healthcare and Consumer Protection of the Republic of Austria.
Dear Professor Carico, dear participants of the Congress of Vienna, it is a great honor for me, dear Professor Carico, to look back on your eventful life and to point out what your research already means and will mean in the future. You grew up and completed your doctorate in Hungary, accomplished an academic career at American universities, and you're the mother of a 2-time Olympic champion and 5-time world champion in rowing. And most importantly, and what no one else could claim for themselves, you are the mother of mRNA vaccines. A lot can be said about your life, your career, and your scientific excellence, but I would like to comment on what you have achieved with just 2 words, which are often neglected, especially in stressful times. Therefore, I would like to say from the bottom of my heart, thank you. Thank you, Professor Carico, for the unwavering spirit, of your research that you have demonstrated over all these years. Thank you for always believing in your scientific work and never losing sight of your goals. Thank you for your achievements in realizing a vaccine that helps to prevent COVID-19 cases and deaths, despite all the unpredictability in the development of the pandemic. You have laid the groundwork for us to confront the virus with a powerful and effective weapon, namely mRNA vaccines, that help revolutionize vaccine and drug technology. For mRNA, the technologies are not only the basis for vaccines against COVID-19, but will also be used in the fight against, for example, cancer, strokes, and cystic fibrosis. We are thus entering a new era in medicine, because thanks to mRNA technologies, we can protect human health faster and more efficiently. For this, dear Professor Carrico, from a public health perspective, but also on behalf of every individual whose life you have improved or even saved with your achievements and will continue to save, you deserve all our thanks. The fact that you are being awarded the Golden Arrow for your life's work is, of course, only a symbolic recognition of this, and probably one of many awards that you are rightly receiving at the moment. But as a symbol, this award suits you as a laureate. Your achievements render possible an era that points to the future from an extremely challenging situation for our health and for our lives as a whole. Thank you.
Thank you, Katharina Reich. Now the final laudatory speech will be given by Eva Kondoroshi, recipient of the Palsom Prize 2018 for Chemical Ecology and member of the Group of Chief Scientific Advisors of the European Commission.
I think there is no doubt. I think everybody knows that Kati is the best non-scientist in the world in these days. She appears on the front page of many magazines. She is on the painted wall in many cities, so she is known by everybody. But I would like to come back a little bit and see what was the history and how she achieved this one. Actually, it was not a good idea to come here. Okay. So she was born in Hungary, and she grew up in a small city in Hungary. Her father was a butcher and the mother is a bookkeeper. She studied at the University of Szeged, and when she got the diploma, she started to work as a PhD student at the Biological Research Center in Szeged. Already this center at that time was a miracle that this existed. It was in the middle of the 60s when the Communist Party gave the green light for the creation of this center, and this was a new modern biological center. This was supported also by the United Nations developmental programs, and they hired the best, most talented young people to work in this center. So the average age of the center when it was opened in 1973 was below 30. At that time there was money to have famous scientists who came to Szeged and helped mentoring of the young people. Kati joined the Biological Research Center in 1978. This was already the time when the UNDP support was over and there was much less money to work and to have chemicals what is needed for the experiment. So this is the time when we started to work together with Kati. And we knew that we cannot order the chemicals. And I hated to work with animals or kill them, but Kati, being a daughter of a butcher, was not afraid to take the brain of the cow and start isolation of phospholipids from the brain. So this was a period when we had to learn how to do research without really support and do everything by ourselves. So we learned that that impossible does not exist. So we have to find a solution. So with Kati, we did the work together, and we managed to publish a paper in '85. And this was the time when really there was no money at all for research and even maintaining the stuff. And Kati had to move, or decided to move, to the US. So she started as a postdoc at the Temple University, and from '89 she became a staff member at the University of Pennsylvania. This was the time when she really concentrated how to use mRNA-based for gene therapy. She applied for many, many grants, and all her grants were rejected. So this was really a very, very bad time for her, and perhaps many other people without so much convictions would have left already the field and would started something else. So one reason why her grants were rejected, it was the fact that messenger RNA was very toxic and raised a very strong inflammatory response. So due to the lack of grant support, she was not going towards the full professorship. She was demoted, and she had to work in very miserable working conditions. Her lab were taken away. She had no equipment, nothing. So we know already that later in 2013, she joined BioNTech as vice president and promoted as senior vice president, where she had the opportunity really to do the research. So when she started to work with the messenger RNA, actually, it was already known from 90s that you can inject messenger RNA, and this can provoke protein production, but they could not use it because it was very toxic. So the difficulties to work with RNA, first of all, it was that it's very difficult to introduce to human cells. This is a big molecule. This is extremely sensitive. It can be degraded very rapidly in the cells, and the immunogenicity. And Kati, by herself and also with collaboration with Drew Weissman, solved all these problems. So first she identified that modified nucleotides can increase the stability of the messenger RNA and increase the translational efficiency. And together with Drew Weissman, they found out that this is uridine which causes this immune response, and replacing this with pseudouridine can make usable the messenger RNA without any toxicity. And then introduction into the mammalian cells, it went very easily, probably based on previous experience as well, using lipid nanoparticles. So, and this was then really the base, and which made the change for the COVID vaccine production. So you can see that until 2020, Kati was not known for the scientific community, except the first recognition which came from Academia Europaea, where she became a member already in 2020, and she got also another prize in US. And you can see the list of the most prestigious prizes what she got in 2021, and you can see that already we are just at the first month of this year. She has already many known, very prestigious awards what she will get in this year, including the Japan Prize, which was announced 4 days ago, and many, many other prizes. And you can see also that she is also recipient, or will be the recipient on 8th of March, the L'Oréal Prize, UNESCO Women in Science. We are debating how women scientists can be successful, which can be behind, and why Kati was so successful. And Kati's all achievements were Based on her strong convictions, determination, commitments, perseverance, stubbornness, and very, very hard work. And I think Kati, not only having this one for herself, but she could give it also to her daughter, or she inherited this one, being so successful Olympic champion, world champion in rower. So this is a wonderful family, very talented mother, daughter, together with a great husband. So, Kati, I would like to congratulate you what you achieved. This is really great, and I am very happy that I know you since many, many years, and we kept our friendship and love forever. Congratulations.
Thank you very much, Eva Kondraschi. And now we're coming to the presentation of the Golden Arrow 2022 winner.
So, thank you very much. It is very nice to be awardee of the Golden Arrow. I might mention that I started and grew up in a very small town with 10,000 people. We lived in a small house. There were no running water, no television, and no refrigerator. So the road to be here was not a straight one. And I am very grateful for all of those people who helped me on my journey. My parents, my mentors, my teachers, my present family, the very supportive husband, and all of the colleagues, fellow scientists, students who helped me. And I hope that it serves as an example for those who might be underprivileged, that they can achieve so much if they put their mind on and work very hard. I am always humbled because, you know, as mentioned here, and thank you very much for the laudation, it was just so moving, and thinking that it is about me, because, you know, the road to success seems like, you know, this is the success. I have to tell you, the success is really that my knowledge was used to save people. So that's the real success. And the recognition I greatly appreciate, and being here, I just say thank you very much. And I keep working and trying to also to serve as an example for women, that, you know, they can stand up for themselves, and then they can achieve so much. And also for those who are immigrant, as I am in the United States, example for that I get an opportunity to work in another country and use this opportunity to learn and work. And so I am very grateful, and thank you very much.
congratulations to the winner of the Golden Arrow 2022. Now we are coming to the second part of the Golden Arrow 2022 Lifetime Achievement Award ceremony. We will start with the jury decision for the special Golden Arrow 2022 winner. And I would like to welcome on stage Katharina Reinwald, Secretary General of the Vienna Congress.
Ladies and gentlemen, distinguished guests, before I'm going to announce the jury statement, I would like to reveal something that is connected to our special Golden Arrow winner 2022. Behind this piece of fabric, there is hidden something that has written sports history. And it is a symbol for endurance, ambition, and success. It was the 25th of July in 2021 when the whole world was watching this t-shirt run by an Austrian. An Austrian who consistently pursued her goals. An Austrian who managed to focus the international attention on Austria. And an Austrian who made an extraordinary achievement. The special Golden Arrow is traditionally granted to Austrians who made an extraordinary achievement and managed to put the international attention on Austria. The special Golden Arrow 2022 goes to Anna Kiesenhofer. Anna Kiesenhofer impressively combines top performances in sports and science. As a mathematician, she deals with complex problems in the area of differential equations. At the 2001 Summer Olympics in Tokyo, on the 25th of July in 2022, she won the gold medal in the women's road cycling race, thereby writing Austrian sports history. Unlike other athletes, she constantly pursued her sporting career alongside her professional career, thereby demonstrating particular consistency and performance orientation. In science and sports, Anna Kiesenhofer is an impressive role model for young people who are at the beginning of their career and are faced with the challenge of pursuing of different areas of interest. Dr. Anna Kiesenhofer, the committee of the Vienna Congress congratulates for the special Golden Arrow 2022. Thank you.
Thank you, Katharina Reinwald. Please now welcome on stage our laudatory speaker, Dan Schechtmann, Nobel laureate in chemistry and vice chair of the advisory board of the Vienna Congress.
Ladies and gentlemen, the history of vaccines changed this time. Something happened this time. When Jenner discovered the first vaccination and then Ian Fleming, they were based on observations. They observed something, deduced the right conclusion, worked on it, and prepared the vaccines. Ian Fleming discovered his discovery in 1928. Penicillin was available to the public in 1945. 17 years after the discovery came the penicillin to be available to everybody. 17 years. This development, this discovery of the vaccination against the coronavirus was not based on observation. It was based on knowledge. It was based on understanding the DNA, the very complex issue of understanding the RNA, and then the messenger RNA, and the ribosome, and the way they work together to produce proteins. It was based on understanding and innovation. And this is why this discovery, this development is so important and so different than anything that happened in the past. Congratulations. This is really a well-deserved Golden Arrow and hopefully many more prizes soon to come. Congratulations. Thank you.
Thank you very much, Dan Schechtmann. And the next laudatory speech will be given by Elisabeth Köstinger, Federal Minister for Agriculture, Regions and Tourism of the Republic of Austria.
Dear Anna Kiesenhofer, ladies and gentlemen, twice as a charm, so here I am again. I'm not sure what you remember of the year 2021, but I spent most of the time washing my hands and wearing a mask. Our next recipient took things one step further. Anna Kiesenhofer decided to win an Olympic gold medal by accident, she still claims. If you haven't heard her story, here is the short version. At age 22, Anna decides to get into cycling while studying for her degrees in mathematics and physics at the University of Vienna and later at the University of Cambridge. Fast forwarded 8 years and the Olympic Games in Tokyo. After a fairy tale race, she finished the road cycling event in first place and 75 seconds ahead of her closest opponent, who, I swear I don't make it up, was completely unaware that there was our very own Anna in front of her. It was Austria's first gold medal since 2004 and the first in road cycling since 1896. You guessed it, we weren't exactly a cycling superpower, plus I'm pretty sure the athlete who won it in 1896 didn't have a PhD. Anna, you taught us a few lessons. Never underestimate an underdog, and never underestimate an Austrian mathematician on a bike. All joking aside, you prepared for the Olympic Games without a team, without a coach. You went your own way while cycling 25,000 kilometers in training. You showed us all that women are capable of making the impossible possible. You showed us that there is not a single way to success, but sometimes we need to walk off beaten path. Most importantly, you all gave us an incredibly inspiring moment of hope and pride during these difficult times. People like you will always be successful in sports and now in your profession as a scientist in Switzerland. On a grand scheme of things, your race might have only lasted a few moments, but in the memories you left us will last a lifetime. Thank you so much.
Thank you very much, Elisabeth Köstinger. I would now like to welcome on stage Werner Kuhn, Director Business Development at SK Rapid. Well, I think we have to wait a little bit further for Werner to join us on stage. May I then ask for Emanuel Klenk to join us on stage, Head of Department for Thematic Programs at the Austrian Research Promotion Agency. Thank you.
distinguished guests, dear colleagues and friends, dear Anna and family. Almost everything has been said, and now I feel very humbled to give you 5 minutes laudatory speech in the honor of such a great person and her fantastic achievements. I am neither a mathematician nor a good cyclist, and coming probably last after such excellent speakers does not make it easier. Nevertheless, when David asked me to address you a few words, I dared not refuse, in part because there are few connections between Anna and myself, and these connections, David, you did not make it exceptionally. We met first in 2008 during your internship, Anna, at the Austrian Research Promotion Agency. At that time, you were studying 5 foreign languages at school. We should always be curious about our colleagues and their hobbies. They might later win a gold medal or a Nobel Prize, or maybe even better, help you get one. Dear Anna, you obtained your master's degree from the Cambridge University at the Emmanuel College, if I'm right, and then wrote a PhD thesis in Catalonia on, well, a topic some of you may have heard of if you work on topology. Although most researchers nowadays would say that teamwork is essential, I guess that mathematics or research in mathematics is often a lonely dive in a very abstract world, expanding the frontiers of knowledge, sitting on a chair, but the brain at full speed. Now you work as a postdoc in my home country, Switzerland, and in my former alma mater at the École Polytechnique Fédérale in Lausanne. I guess the mountains gave you the strength to increase your cycling skills, and I'm sure you enjoy the Alps and the countryside. After being Austrian champion 3 years in a row, you succeeded last year in Tokyo in overcoming your own limits once again, using brain and muscles, sitting on a fast and furious saddle. Just to give you, the audience, a picture of the last minutes, Anna was far ahead of her closest followers, pushing hard, giving the last drops of energy. Maybe she was starting to realize that she was going to win the race. Seeing her beyond the finishing line, lying exhausted on the asphalt and almost crying, I felt deeply moved. Anna has won with determination, courage, and her own strategy. Anna has moved once again the horizon of her physical and mental possibilities with the same dedication, the same perseverance, and the same modesty as before, creating something uncommon, entering the unknown. Innovators are outsiders. They expand frontiers and break limits. Dear Anna, I'm sure you're one of them. You have shown the whole world that you were very able to succeed a couple of times between your PhD and the gold medal to break those limits. I'm poor at statistics. Maybe there is a specialist in the room here or online, but I guess that you will, Anna, make it many more times in your life. You dare to be different, as you state on your homepage. Maybe you're a serial innovator. In a recent radio interview, you said that you were not sure whether the whole effort was worth it, whether the years of dedication invested in study and training could have been invested in a different life. This is a fundamental and tough question for a young person to ask. And in a world where so many people are ruled by certainties, You allow yourself to doubt after having achieved such brilliant results. My greatest respect for that mindset. Dear Anna, you succeeded to be the best, and for that, you fully committed to your objectives. I would like to congratulate you personally for the extraordinary achievements so far and wish you the best luck in life. I'm sure your skills will further expand the frontiers of knowledge in the quest of truth. You represent for me the young generation and my personal hope to shape a better future. And for that, you deserve the special red, golden, I don't know the color anymore, a roll. Thank you very much.
Thank you very much, Emanuel Klenk. Now we are coming to the presentation of the special Golden Arrow 2022 winner, and I would like to ask David Unger-Klein to join me on stage.
Dear Anna, I just want to give you one sentence, never give up, you can reach everything. And we have here die Urkunde, but we are missing the golden arrow. So we have one more to come. And Anna, I would like you please to come on stage. Und nun kommt der größte Fan von dir, Anna. Du kannst die Maske runternehmen, aber du kannst ihn gleich empfangen. Das ist der Leo Unger-Klein, der jetzt mit dem Fahrrad den Golden Arrow bringt. Leo, bitte.
Danke, Leo. Thank you very much for this prize. I'm feeling very humbled to receive it. Thank you also for the nice speeches, especially Emanuel. It really touched me. Yeah, I'm kind of starting to be a role model for people working in science and sports at the same time. And it almost feels a bit strange. I have been hardworking during my whole life, and it won't surprise anybody if I say that in science and in sports you have to be hardworking in order to succeed. Everybody here, I mean, we have so many famous guests here, Nobel Prize winners, famous politicians, scientists, and yeah, some sportsmen, women. Everybody will agree that you have to work hard in order to succeed. But what I want to say is that hard work does not guarantee success. Hard work is necessary, but hard work is not sufficient, at least for having success in the public eye. My whole life before Tokyo was about hard work and not having success in the public eye. I had success for myself. Tokyo was not the first race I competed in. I had so many small goals I was focusing on. I was sacrificing everything for it, and I was often laughed at for sacrificing so much for seemingly small goals. People will judge you whether your goals are worth focusing on. And if they don't seem big enough, they will laugh about you when you work hard. When I don't go to a party just because I have this little race in my hometown. So yeah, Tokyo was another success for me. It was a really big success, but it was not so different from the other small successes I had had in the past. What was really different was the way people viewed me. The perception of the public changed. Suddenly, I was somebody who got invited to events, who is supposed to say something intelligent, who is supposed to do things the right way. It's strange. Actually, I haven't changed. I'm doing things just the same way as before, just as right or wrong. I'm certainly not complaining. This success has opened a lot of doors for me. But I want to always remember where I came from. Just as in science, in sports, the attention goes to a few select people. The attention and also the funding. With this speech, I want to draw attention to all the small goals, to all the people who sacrifice their free time for things that are worthwhile for them, but who do not get this recognition. My Olympic gold medal made me famous, but I do not want it to be some remote fairy tale of a super talented high achiever that normal people cannot relate to. I am not a super talented high achiever. I'm a normal person. And this Olympic gold medal stands for all the small successes I had in the past before the win that made me famous. And it should stand for all the other small successes that other people have, for all the sacrifices that go unnoticed. It really doesn't matter what goal you are preparing for. It sometimes pains me when people now approach me and they say, okay, I'm not like you, I'm only a hobby cyclist, I'm only preparing for this or that race, I'm only a student. There is no only in a person, there is no only in a goal if it is important to you. It is not about what is written on a medal that determines success. Doesn't matter if it says Tokyo or Olympic or what color it is. This is not what determines success. It is the obstacles that you overcome towards your success that are important. Thank you.
Congratulations to our winners of the Golden Arrow 2022. Now it's time for a break. Please may I ask you all to be back again in this room by 3:20 so we are ready to start on time. Thank you.
Hallo?
Welcome back, everyone. Our next focus topic is on the future of our health under the title Together Against the Virus. Before the panel discussion starts, we have the privilege to hear one video message and 6 introductory notes. First, I would like to open our next topic with a video message from Tedros Adhanom Ghebreyesus, Director General of the World Health Organization.
Dear colleagues and friends, the COVID-19 pandemic has upended our world. Governments have faced many difficult choices, but many of the most important choices were made well before the crisis itself. The choice to invest in primary health care and public health infrastructure, the choice to put science at the heart of public health policy, and the choice to build trust with communities through a strong social contract. The immediate challenge we face now is to end the acute phase of the pandemic, including by ensuring equitable access to vaccines. That means vaccinating 70% of the population in all countries by mid-2022. While COVAX has delivered more than 1 billion doses of vaccines, we're still far from reaching our goal. 34 countries have still not been able to vaccinate 10% of their population, and 86 have vaccinated less than 40%. And of course, vaccines alone will not end the pandemic. Many countries also need diagnostics, life-saving therapeutics, including oxygen and support for vaccine rollout. But by working together in solidarity, we can end COVID-19 as a global health emergency this year and build a healthier, safer, and fairer future for all of us. I thank you.
Thank you very much, Tedros Adhanom Ghebreyesus. Please now welcome on stage Heidi Stensmyren, President of the World Medical Association, Chief of Women's Health and Health Allied Professionals at Karolinska University Hospital in Stockholm, Sweden.
Thank you so much. Dear distinguished guests and participants, while it's easy to dwell on the turmoil of that COVID has caused worldwide, there's a glaring bright spot amid the chaos. Our scientific community, as you've seen here today, capable to make remarkable breakthroughs. Excellent scientific work as the production and use of mRNA tools has given us the COVID-19 vaccines. The revolutionary breakthroughs can contribute to change the face of healthcare as we know it. The willingness of companies and authorities to act fast and take on risk has been a precondition to make it happen, though, and we should not forget about that. Collaboration achieved high speed in the translation of scientific evidence into health policy and bringing vaccines to the market, a proof of society's high capability to adapt. Just as collaboration between research, community, healthcare, and politics to secure healthcare to and the safety of the population are key to success, here also lies the challenges. Sufficient healthcare system is a pillar in a well-working society. WHO has shown a clear correlation between spending on healthcare and countries' development in meaning of growth of the economy. As you heard Tedros here say, access to healthcare, access to primary care is maybe one of the best investments we can make in society. As a young medical student, I read the book The Plague by Camus. I was seeking the medical expression of a plague. But, as you all know, it was much more about the social effects, the context between people and society, how lockdowns were accepted or not accepted, how trust and distrust affect individuals' coping capability, and also affect the development of the epidemic. Handling then a pandemic is about, well, much more than to treat an isolated medical disease. Knowing that a high rate of vaccination in the population globally and a resilient healthcare sector are needed to overcome this crisis and be able to meet the next crisis, the obvious question is what needs to be resolved. To succeed to achieve a high vaccination rate, some key factors are needed. Securing access for all, which means scaled up production, of course, efficient distribution structures, we can do that. Secured finance, so that everyone can have access to vaccines. It's possible, but it will need global collaboration and the insight that the pandemic is a global responsibility. So, and I pointed out global collaboration. The world society has yet not succeeded. We're not there yet. Hopefully, initiatives like Gavi, the vaccine alliances, co-leading WHO COVAX, will grow stronger and in the future have both mandate and funding enough to provide sufficient vaccines globally. The global institutions are important and needed, and we should all support them. But none of this will have the aimed effect if we cannot build trust in the population to be vaccinated. And trust cannot be forced. Trust needs to be earned. And it needs to be earned in every generation, in every society. We need trust in institutions, trust in political systems, trust in research and healthcare. And we have a big responsibility, the medical society, in contributing to trust. So building trust is one of our most important challenges. I mentioned trust in the system, but also trust in science, trust in the governing bodies, trust in decision makers, trust in physicians. One important touchstone in trust in research is that it's being performed in accordance with a well-grounded ethical framework. Securing the implementation of the Declaration of Helsinki in national and global framework for research is crucial. The declaration, drawing up fundamental ethical principles for research on humans, is important for the legitimacy of medications, the legitimacy for vaccines, and for the healthcare provided. This counts for the society as a whole. The legitimacy of institutions, let it be parliamentarian institutions or organizations, builds on the steadiness of formal rules that are agreed upon and broadly accepted. It is an individual responsibility to participate on the agreed upon rules as well. Further, achieving sustainable healthcare with sufficient capacity is not only a big future challenge, it's a daily challenge around the globe. Sufficient workforce as well as sufficient support of key components and medications is needed. As you have heard or seen in the news, hopefully, and many of you have participated in, health professionals and other health personnel work to their limits and beyond the limits, with great personal sacrifices. WHO estimates that far more than 100,000 health workers died from COVID-19, and they are still out there in the front line, providing care today, every minute of the day, every day around the year. Around the globe, thousands and thousands of healthcare professionals provide care to patients falling sick to COVID-19, often with the consequence of being stigmatized and attacked. And they ask the population to get vaccinated. There is evidence of high rates of PTSD amongst healthcare professionals. In some reports, 20%, in others up to 30% of the health professionals. suffering from this. A significant portion of healthcare professionals plan to leave the healthcare sector totally in the next 1 or 2 years. A study from the US showed that as many as 20% of the physicians plan to leave the healthcare sector. So, what is needed? To be resilient, we need to transform the way we provide care, because healthcare is high-tech. Medical diagnostics, therapeutics, often provided in a less flexible organization. We need to be far more agile and capable to scale up when needed. And this transformation of healthcare is essential. To provide healthcare at the same level as we do today, but provide it in the same way as we do today, we need to recruit whole graduation classes to healthcare system. This is not sustainable. That's why we need to transform the way we provide healthcare, develop and implement new technologies. And even here is trust needed. Healthcare needs to trust technology and open up and have confidence enough to work in new ways and implement the technology and new solutions. The most successful healthcare facilities and hospitals around the world coping with COVID-19 were able to transform, to change the way they worked, to scale up the needed facilities, like the intensive care unit. Thereby, we will need to rethink the role of healthcare, the social structures for our economies. Lack of capacity and crisis preparedness in healthcare is costly, not at least because of lockdowns and other restrictions. And we need to invest right in healthcare. In most developed countries, around 10% of GDP is spent on healthcare. But the question is then, how can we invest in a way that we secure a better utilization of this amount that we spend? Investments in new technology and implementation of technology is needed. And this is also when it comes to scalable and adaptable, as I told you. Regrouping the way we work to adapt to the current need and not the need of yesterday. We also need to secure our supply. To rely solely on a single production unit for a vital component made in one country is not sustainable. That's vulnerable. We might need to build up regional production of vaccines, also production lines. The pandemic is global and needs to be handled globally, but we do need to provide vaccines regionally. So, another point I would like to make here is when I'm into costs and investments, lockdowns of school is extremely expensive and long term. The economic benefit of securing schools and education for all children are well known and should have been better regarded in many situations. It's a long-term effect of closing schools. So we have talked earlier today about investments in education, and it's maybe one of the most important things we can do. So, healthcare is a common good and common obligation. Defining healthcare as an investment and not only a cost will make is a good solution. Universal health coverage needs to be available for everyone. And as I said, it's a global dimension of that. Pandemics are internationally by definition, they regard no borders. Pandemic preparedness must have a supranational character and resourcing. A pandemic treaty is now being proposed to the WHO, and it might be a step in the right direction. Because health is not only it's not only local business, it affects us globally. We also need to regard the livestock, the agriculture, because they are big consumers of antibiotics, for example. So, having spoken about the healthcare system, capacity, capability, scale up, production, I come back to trust. Because it flourished misinformation, not the least about COVID-19 and vaccines. and we must make sure misinformation will not have a significant effect preventing people from being vaccinated. Because COVID-19 and misinformation about vaccines is ubiquitous. The new study, Rise and Fall of Rationality in Language, published in the Proceedings of the National Academy of Sciences, found that facts have become less important in public discourse. Over the past 40 years, public interest has undergone an accelerating shift from collective to the individual, from rationality towards emotion. We are living in a post-truth era, an area where baseless beliefs and subjective opinions are given a higher priority than verifiable facts. The report assesses the public awareness of and belief in a range of more or less well-known myths. And I will not mention them here, but you probably hear them in the news. It's about diseases and the vaccines to prevent the disease. An alarming high number of the population believes in them, despite no lack of correct information provided by numerous researchers and solid public institution, democratic chosen, the common rules. So vaccinations and compliance to regulations and restrictions depends on trust, and that's why building trust is key. So with those words, I'll thank for my time here and leave the words to you again. Thank you.
Thank you very much, Heidi Stensmiren. Now I'm very interested to hear what our next speaker has to say. Please welcome Ottmar Kläuber, Secretary General of the World Medical Association.
Thank you very much, ladies and gentlemen. I have the pleasure to be here for the second time at this rostrum and to address you, the community of the Vienna Congress, virtual, hybrid, far away in part, and nevertheless together again. It has been a difficult time, and we are now here to discuss and to learn how we can do better after the pandemic. It was a time during the past 2 years, and I guess that's true for most of us, with ups and downs. The ups we have celebrated in part today, with a remarkable work of scientists, but also of companies, of health professionals, of doctors, of nurses in caring for their communities, caring for the sick, but also caring for the population by providing vaccines that are highly effective and safe and have already very likely saved millions of lives. Ups because it was in an extremely short time that those vaccines have been developed. And I must confess, when I heard in 2020 from some of my colleagues in late summer, in early fall, they told me it will work, we will have a vaccine against COVID-19. It was really a moment of relief. It was also a moment of belief in science that this can work. And to me, it was also very clear that this was a great, huge step forward, not only for fighting COVID-19, but also for providing vaccines for other diseases, providing medicines for other diseases with this new technology that has been proven to be effective and working. But it has also been a time of downs, of very sad moments, of colleagues, of friends lost. It has been a time which was disappointing when we saw that countries were falling into nationalism and egoism, and very suddenly cooperation did not work, even on the European level. A time where we had to learn that we are able to produce vaccines for billions of people, but that at the same time, billions of people were not served. We heard Dr. Tedros saying that equity is important. And equity is important especially during a pandemic, because a pandemic, and that is what the word says, is everywhere. And as long as we don't find it everywhere, we are not getting rid of it. Success will be to reach equity in providing resources for healthcare, including vaccines, including PPE, to all those who need. And that, I'm sorry to say so, is something where we have not been very good at. Some may say we failed. Being prepared for a pandemic, quite obviously, is not something that can stop at borders, but has to lead to international cooperation. Yes, I do understand health ministers had to care for their own countries first. That's what they have given their oath for. That's what their populations expect. But that, again, is not enough for fighting a pandemic. When we are now talking about an international treaty on pandemics, this may be a way forward in order to prepare us all much better for a next pandemic. Giving the institutions like WHO a chance to really act directly, very openly, and with force against a pandemic will be necessary to do better next time. Money is important, but the willingness and the cooperation of the states and the government to do so as well. Second, our communication in the beginning of the disaster, and to a large extent during the disaster, was not good enough. Heidi Stansman said it, we have to work more on building trust. And building trust in the first place is a matter of communication. And despite the wonderful achievements in developing the medicines, the vaccines for COVID-19, let's face it, we have not been able to convey the message good enough. In many of our countries, there are 10, 20, 40% of people not vaccinated. Even so, the vaccine is available freely to them. Even worse, some pay money to have fake vaccination certificates in order to show them when they are required. Again, at a time when they can have a vaccine for free easily. So, when it comes to learnings from the pandemic, certainly to improve our communication is one of the key issues that we have to look for. And it is something where we still have to achieve a learning curve in how to build better trust with our populations and trust in science and belief in science. Third, we need a better preparation in our healthcare systems. We need it at home, even in the rich ones, because we have seen that many of our even very elaborated healthcare systems, including those here in Europe, have not been able to have enough surge capacities for the pandemic. Second, we need to build better healthcare systems in most of the countries. The key word here is universal health coverage. And that is something we have to achieve in all countries of the world, not only in the very rich ones. This is a political challenge, but it's also a challenge for us as physicians, as organizations of physicians, to help to achieve this. I think for those who have been working very hard in science, in clinical service, be they nurses, physicians, scientists, or others who have helped to work on this pandemic front, I think we owe it to all of them to do our best to learn from this pandemic and next time to be much better prepared. I thank the organizers of the Vienna Congress, the board, the advisors, David, for the work they have been doing and for focusing the attention on our dealing with the pandemic. And I hope very much that once we come together here and can say we really have learned from this pandemic and now we are doing better. Thank you very much.
Thank you very much, Ottmar Gläuber. And now our next speaker is joining us online. Please welcome Leonid Eidelman, former president of the World Medical Association and of the Israeli Medical Association.
Mr. David Ungerklein, the initiator and organizing of the Vienna Congress. Thank you very much for a great job that you are doing. Dr. Heidi Stensmieren, the president of the World Medical Association, and Dr. Ottmar Gläuber, the Secretary General of the World Medical Association. Thank you very much for participating in this very important meeting. Dear organizers and participants, ladies and gentlemen, distinguished guests, 2 years ago, when we have been in the Congress meeting in Vienna in 2020, we have heard about the new virus, and we didn't know what the future prepares for us. Since then, in Israel, as an example, the first confirmed case was found in 27th of February, and the second wave started at the end of May. The third wave was Alpha wave, it was in December. And then the vaccination of Pfizer vaccine started in Israel. Since then, we probably are the only country that vaccinates only with Pfizer. And our case study is something that should be studied. The fourth wave, Delta one, came in April, and the fifth wave of Omicron at the end of November and during the December and January. And now we are actually in the peak of this fifth wave. It's raised very rapidly, and in Israel we have more cases per million than in any other country. We have 99% of our cases is Omicron, like in the United Kingdom, United States, we don't have a lot of Delta now. And we have about 1,000 severe cases. Of these severe cases, you see, if you are vaccinated and you are under 60 years old, your chance of being severely ill is very low. If you are 60 years old and above, your chance is higher. But your chance is much more higher if you are vaccinated in the past, you have outdated green pass, and if you are not vaccinated, this chance is huge that you will be severely ill. In Israel, we have started very early and reached more than 50% very rapidly. But since then, we have a plateau regarding the rate of vaccination. Actually, if you calculate on those who are eligible to get vaccination, one dose got 80% of this eligible population. 2 doses, 73%. 3 doses, 53%. And 4 doses, 41%. For 4 doses, for the 4th dose, population that is eligible, it's medical staff and those who are above 60 years old. And you see also with booster, after 2 vaccination came the first booster. And then, as I told, we have for a certain population, the second booster, the 4th vaccination. And you see, we are very high in comparing with other countries in new hospital admissions. But if I take data, and a very reliable site, our World in Data, unfortunately, they have been mistaken by showing that our number of new ICU admissions per million is so high. We don't have so many ICU beds. It's actually severe cases. Severe cases are ones with oxygen saturation in their blood less than 93%. In our ICU, we hospitalize patients who are ventilated, getting artificial ventilation. And in our ICU patients with COVID-19 per million, you see we are somewhere in the middle. Somewhere in the middle. Daily confirmed cases are going up. Deaths per million, we are somewhere in the middle. And excess mortality, which is probably the most important parameter that is measured in all the countries. We are now going even down. It's about 0%. I'm asked about stringency in Israel. It's about also in the middle of all the countries. And now it's even going down because isolation in the schools are now stopped. And I'd like to show you two studies of many that are produced in Israel. And one is a study of non-hospitalized patients. And conclusion is the vaccination with at least 2 doses of COVID-19 was associated with a substantial decrease in reporting the most common post-acute COVID-19 symptoms. Long COVID is much less frequent after at least 2 doses, bringing it back to baseline. Our results of this study suggest that in addition to reducing the risk of acute illness, COVID-19 vaccination may have a protective effect against long COVID. And I would like to address the letters that was written by physicians from our country was sent to the editor of New England Journal of Medicine, and I would like to cite it. The importance of third vaccine dose is clear. Actually, the decision of our government for the booster was very reasonable because it passed a lot of time passed after we started. We started in December, the first vaccination. It's the third vaccine dose on the higher neutralization efficiency by a factor of 100 against the Omicron variant after the third dose than after the second dose. Boosters, the third dose is a booster dose. However, even with 3 vaccine doses, neutralization against the Omicron variant was lower by the factor of 4 than that against the Delta variant. For the Delta variant, it was very effective. And you have seen the publication in media, not in scientific media, because no work was sent to scientific journals, not yet. The data was not analyzed and was not peer-reviewed. But you see, it caused the very high interest in general media. Israeli trial, world's first, finds 4th dose not good enough against Omicron. The question is about the second booster. And I would like to bring to your attention the case study based on the events in my department. In my department, we have 70 physicians. Mean age, 41 years. All are vaccinated with 3 doses, and some physicians above 60 years old with 4 doses. During the previous waves, we had around 7% of them confirmed with COVID infection. During the 5th wave, when I prepared this presentation, we had 21%. Today, I can report you that we have above 27%. It is close to 30% of our physicians who were infected with Omicron. But no one needed oxygen and no one needed hospitalization. All those physicians who were infected and confirmed with COVID-19, actually with Omicron, passed this disease quite easily. And we see now in Israel the R coefficient, effective reproduction rate, which shows how many new cases one patient can infect. It is going down, and it is about 1, which means that the situation is stable. Actually, it was 3 days ago when I prepared this presentation. Today, I have got the information that it's even less than 1. So we hope that this wave will be rather short and will go down in the next 2-3 weeks. So my hope is, I believe that Omicron, also the subvariant of Omicron, the next one that was discovered recently, is very infectious but less pathogenic, and probably it causes the natural immunity. And my hope is that it will be the end of this pandemic, and the next year we can meet in the Vienna Congress in person, all of us. Thank you very much for organizing and for the opportunity to present our data. Thank you very much.
Thank you very much, Leonid Eidelmann. Please welcome our next speaker on stage, Stefan Ludwig, Institute of Virology, Center for Molecular Biology of Inflammation, University of Münster.
Thank you for the introduction. So as you heard, I'm a virologist, and virologists are a species of scientists nowadays that have evolved to be bringers of bad news. And I'm afraid that I have to start my presentation here also with bad news if it comes to describing the current situation of the COVID-19 crisis in the eyes of a virologist. So when I was here last year to report on new data on SARS-CoV-2 findings, I wouldn't have expected that I will be here at the same place and reporting the same problems again. And that's the sad news, that we are still in the middle, or not in the middle, but we're still in that pandemic. And there is a high number of infections worldwide in the moment, which is not good. So, and this is a real severe and fulminant pandemic. We will see that pandemic showing up in the history books in the future, lining up with pandemics like the Spanish flu in 1918 or the Asian flu in 1957. It's certainly not as severe as the Spanish flu, but it can easily outcompete the flu pandemics of 57 or 68. So we have a real bad pandemic we face, and we have to accept that this is a natural disaster, and this kind of natural disasters come with a cost. And we learned a lot today already, not only about the costs of lives and people who suffer from long COVID, but also the burden that was occurring due to that pandemic at the social level and also in the interaction of states. So I'm starting with that negative result, since there are still many people out there saying that this pandemic is not really so severe and that some measures are overtaken. So I would not agree with that. So we still have to do something to overcome the pandemic. Starting with these negative statements, there's also a lot of positive things to say. And one of the positive things is the science and research on COVID-19. Actually, there are hundreds, if not thousands of laboratories worldwide who focused their work on COVID-19 and SARS-CoV-2 and wanted to study the replication of the virus, to learn more about that and to find measures to fight the pathogen. Thus, we know already an awful lot on the virus, and it's no doubt that the exponential gain of knowledge, there's no other example on an infectious disease in the past where we saw that in such a tremendous way. So we know already a lot, we do not know all. The most visible sign of this enormous power of research is, of course, the very, very fast development of efficient vaccines in absolutely record time. We really have to be aware that this is an absolute fantastic success story, despite all these somewhat absurd discussions about efficacy and safety. And what we saw here is also the introduction of a new method, which is now used on a broad scale to find an infectious disease. This has caused in some areas of the society the notion that this is all very new. But we learned today from Katalin Karikó that, of course, the development of mRNA vaccines has a long history already. So, and by now, billions of doses of that vaccine have been applied to people, and probably by that it's the most tested vaccine ever in the world. And although there are side effects, yes, there are recorded side effects, but they are rare. They're in the 0.00% of cases rate. And this stands in clear contrast to that what you see with infections. If you see, or if you look at studies on the incidences of long COVID in infections or infected patients, you would see up to 40% of people having these long-term complications. And these complications are not well understood yet, and they are not very well treatable. Thus, to summarize, and I line up here with all the previous speakers, the vaccines against COVID-19 are safe, they are efficient to prevent severe disease, and vaccination was the game changer in the COVID-19 crisis. And for the record, there is no medical reason to not get vaccinated for an otherwise healthy person, full stop. No further discussions needed at this point. Will vaccines be the only way to fight pandemics or epidemics in the future? Well, probably not. To fight a disease like COVID-19, an acute disease, we also need drugs. We always have to stand on two pillars. One is vaccination, for sure, but we also need drugs to heal patients which are in intensive care units. And what we also learned during the crisis is while the vaccines have been developed in record time, it is much harder to develop efficient medications against COVID-19. And why is this? Why we do not have really efficient drugs yet? So the reason is that COVID-19 disease is among these hyper-inflammatory diseases which actually occur in several phases. And you can distinguish three phases, at least for the COVID-19 disease. One is the early phase dominated by the virus. Of course, the virus infects cells in your body and spreads in your body and does damage to your cells and your tissues. However, then in the intermediate phase, the virus gradually goes down and the damage caused leads to something which is called a cytokine storm. So actually your body reacts with the expression of cytokines which actually is meant to fight the virus. However, these cytokines are then produced in such an enormous mass that they would be directed against your body. And then in the third phase of the disease, this cytokine storm, this hyperinflammatory response takes over. And the virus actually does not play a major role anymore. And if you look at these causes of the disease, you can understand while drugs you may all have heard of, like remdesivir or molnupiravir, that both target the virus directly, wouldn't be very effective and weren't very good in rescuing people from death and in treatment of highly severe cases of COVID-19. There is now some light on the horizon. There's a new drug, which you may all have heard of, Paxlovid, which is also a drug that targets the virus directly. The clinical trial data so far look good. However, experts also say that this drug has also to be taken very early on after infection and would probably not be very good in severe and late-stage patients. So I still lively remember when I was here last year when we discussed on that podium with Rolf Zinkernagel, an immunologist and Nobel laureate, which you all know. And he said, I quote, "I wouldn't bet my money on direct-acting antivirals against these hyperinflammatory and very acute diseases such as COVID-19. So, and I completely agreed with him. So can we do better? Can we do something different? And allow me to propose here that we probably need a paradigm shift in the development of antiviral agents, or let me better say anti-infective agents. We should not only have an eye on the pathogen and fight the pathogen, but we should also keep in mind the whole course of the disease and also look at that, what happens at later stages in terms of the hyperinflammatory reactions. That would mean that we may need drugs or drug combinations that both target the virus directly and can rebalance a hyperinflammatory response. Another simple and so far, I have the feeling, neglected fact is that every virus needs cells in the body to replicate. And that means that the virus exploits cellular factors for its replication. So why not simply targeting the cellular factors which the virus misuses and thereby indirectly fighting the virus? So this all looks like a very future visions. However, there are already drug developments on the horizon that meet all these new directions and new strategies. And I will only mention one example among many others which exist because we worked ourselves with that drug. The drug is called zapnometinib. And zapnometinib targets a cellular enzyme that is required for the replication of SARS-CoV-2 viruses, but also, interestingly, for other viruses such as influenza or hantaviruses. At the same time, however, zapnometinib also blocks signaling pathways that are involved in the induction of cytokines, and thereby zapnometinib can reduce the cytokine storm in late phases of diseases such as COVID-19. So these drugs are now under, or some of these drugs are now under clinical evaluation. Zapnometinib just undergoes a phase 2 clinical trial, so it still has to show in the clinics whether this strategy really would work out. But I would not hesitate to predict that these kinds of drugs may be game changers for future outbreaks. And maybe then also people like Rolf Zinkernagel would bet their money on that. So besides this kind of outlook on future antiviral drugs, what else will the future bring for COVID-19? And there are some reasons to believe that there is light on the horizon and that we are now in a critical turning point in that pandemic. At one hand, this is due to a variant called Omicron. The Omicron variant, as we just heard, seems to be less pathogenic and spreads very rapidly. So we have high numbers of infections, but not very high numbers admitted to ICU units. And at the same time, now there is a decent amount of the population of the world vaccinated. So these infections also meet vaccinated people or people that have recovered from the disease. So in these people, the virus cannot be so aggressive anymore. So there might be good reasons to believe that we are on the first step in the transition of a pandemic to an endemic phase. And a good guess would be that maybe the SARS-CoV-2 virus will line up with other common cold disease viruses like common coronaviruses like the flu or so on. And then you might need a vaccination every year to cope with new variants. But having vaccines on the one hand and having antiviral drugs on the other hand will take away the threat of these pathogens. So one thing has, however, to be or is mandatory to reach that goal. And this also has been mentioned several times. A pandemic is a worldwide issue. And we also need to gain access to countries which are under-resourced in terms of vaccines and antiviral drugs, and then we will be able to cope with the disease. Thank you very much.
Thank you very much, Stefan Ludwig. Now please welcome on stage Alexander Biach, location lawyer of Vienna and Deputy Director of the Vienna Chamber of Commerce.
Thank you, ladies and gentlemen. Healthcare will be the game changer in this pandemic situation. I'm pretty much convinced about this fact, and thank you, Mrs. President Sternsmeier, about the idea of concentrating on trust, because I also believe that concentrating on the power of trust will be the exact game-changing moment in this pandemic situation. Disrupted international supply chains, a damper on the tourism industry, the grounding of our aviation. Well, all this was the experience throughout the last 2 years, an extremely challenging time for states and economies. So it is the right time, and let me thank you for this initiative, for this Vienna Congress to face all these challenges in future times. Well, smarter heads than I will talk about the medical impacts, but as Vienna location advocate, I would like to focus on the questions that how to solve the global challenges related to this pandemic situation, and connected with the aim of reopening our world, our city of Vienna, for citizens, businesses, tourism, international congresses, well, open the city again for the world. And I believe that investing in healthcare systems may lead to the change in this actual pandemic situation. So just for the first step, let me point out the 4 challenges for the trust situation of the society and economic situation. First of all, the pandemic led to a trust problem for the political situation. Within these 2 years, we lost part of the trust in the management of the healthcare system, and management mistakes where they opened the gateways for disconnected and protest situations all around the world. Doubts about the government ability to act grew, and this makes it for political systems extremely necessary to challenge these doubts and protests. Otherwise, you open the possibility for far-right forces to stir up concerns and fears. Better political management, especially of the healthcare system, will lead to the necessary trust in political systems. Second, the Spanish Elcano Royal Institute stated that within these 2 years, forced by the different actions taken, we can realize some trend of de-globalization, which means that through all these fragmented actions taken by different states, continents, well, even here in Austria, we have sometimes 9 different actions taken to face or battle the pandemic situation. Through these fragmented actions, the trust into institutions was damaged. To counteract this deglobalization might be one of the common tasks for healthcare systems, for example, within the European Union. Third, we have to regain the trust in social stability. Yes, inequality grew with this pandemic situation. The German Konrad Adenauer Stiftung, for example, speaks about the necessity of a renewed social contract between industrialized and developing countries. Well, I believe that closing this gap can work by establishing a broad access to healthcare systems worldwide. And the fourth trust problem we are facing is the economical trust, or the economical freedom. To battle this pandemic situation, it took a lot of debts we built up throughout the last years. Debts to fight all the difficult situations for businesses, to save jobs, but all these debts led to a hill of debts, to a hill of financial obligations. We built these hills, and these hills, they weaken our economy, they bind the economy, they throttle it. Hills of debts that also lead to the question, how can we reduce these hills? Well, there are 3 ways of reducing debts. The easiest way is to pay them back or to cut them. This is a difficult way in the moment, actually. We have the possibility to answer to this debt situation with the growth of economy. So it will be necessary to upswing, as is also pointed out in this Congress, the economy. What is a dangerous way, but what we can experience at the moment is to choose the way of inflation, because inflation reduces the value of debts. But this is a way which leads, and we can experience it, take a look at the energy prices at the moment. The way of inflation, in order to battle the pandemic economical situation, will lead to, again, to equality and poverty. So it will be the question of wise investment in order to gain yields, to find a way out of the economical situation. This will be the hill we climb. So we face not only health aspects in this pandemic situation, we will have to give answers on social, institutional, political, and economical aspects. And I believe that investing and strengthening the healthcare sector will be the answer and the game changer to these 4 challenges. Let me give 3 examples we were working on, or at the moment are working on. Trust through investment. I believe we have a project at the moment running by building up research infrastructure here in Vienna. A center for precision medicine, as we try to build up in order to give exact individual answer to medical questions. It is not cheap to build this up, but this investment will find a way of leveraging this investment in form of a really high yield in order also to solve some economical problems, and it will strengthen the belief in research and medicine and gain the power of science again. A second example we can give is the need for strengthening the institutional trust. What we need is a European Union-wide coordinated healthcare policy, like building up the sometimes mentioned HERA, the Health Emergency Preparedness and Response Authority. But we have to build this HERA project up together, not somewhere in the backyard of Brussels bureaucracy offices. All the countries and all the people concerned with the need of emergency authority should be included. Build up the health coordination, then we build up the trust by coordination. And a third example to regain the trust is the so often mentioned free access to healthcare security systems. Like, for example, the free PCR testing system we built up in Vienna. It is a project organized by the Chamber of Commerce and the city of Vienna. And it's an easiest way of offering everyone living or visiting the city free access to PCR testing. It's easy. You test in front of your smartphone, drop it at over 600 grocery stores all over Vienna. You receive the PCR result within hours on your smartphone again, and then you can enjoy a worry-free stay or movement through the city for the next 48 hours. This project, we call it Alles gurgelt, everyone goggling, is used every day by about 500,000 people in this city. Well, and it gives back some way of being isolated, showing a way out to battle the virus, because using the PCR technology helps to isolate infected persons before they are being contagious. This project also created thousands of new jobs, and this is the third part, trust by access. So let me come to the end of this keynote. Yes, we can overcome the pandemic effects on society, on politics, on institutions, on the economy by trust. By trust, but trust has to be specified and realized. Therefore, we need the healthcare sector. Healthcare will be the game changer. Let's realize the trust by investments in, coordination of, and access to the healthcare system. Thank you.
Thank you very much, Alexander Bier. Our final speaker will join us online. Please welcome Thomas Südhoff, Nobel Laureate in Physiology or Medicine.
Good afternoon or good morning. I have to say it's always a strange experience to participate in meetings via a Zoom link. And so let me first express my gratitude for the opportunity to talk to you here today. I wish I was there in person. I cannot tell you how much I wish that was possible. And it's an honor for me to be part of this Congress. Let me start off by explaining a little bit my own relation to COVID-19. I'm a scientist. I'm sitting in my office at Stanford, but my lab doesn't directly work on COVID-19. In fact, I'm not a virologist. We use viruses, but that's all. However, I work for several drug companies in various capacities, most importantly, probably for Sanofi. And as a disclosure, as you all know, Sanofi is developing a COVID-19 vaccine with some difficulties in between. I serve on the board of directors for Sanofi, and I have dealt extensively with the problems associated with that vaccine. I can only tell you that I strongly believe that we need more vaccines, that we need better vaccines, even though the vaccines that we already have are fantastic, and they are incredibly successful in 1,000 ways. I have very little to add to what previous speakers have said. In fact, I agree with everything that was said. It is really important to emphasize in particular the role of the science here. So instead of repeating what others have said so much better than I could ever express it, I would like to focus on two particular features of this devastating pandemic. And these two features have one thing in common, which is that I think that, as already pointed out by previous speakers, there's a breakdown in trust in our societies. And part of that breakdown, I believe, is the fault of scientists like myself, of so-called experts who haven't been able to always communicate lucidly the actual situation, and sometimes maybe even a little too bound by previous ideas, because I think that COVID-19 has changed a lot of our assumptions about infectious diseases. So the first thing I wanted to discuss is how different COVID-19 really is from any other pandemic that has previously occurred. When initially emerged, I believe most people anticipated this to be a devastating, plague-like viral disease that would come, but that eventually would pass, that would be combated by vaccines and drugs, and that once over would be over. This hasn't happened, as we all know. We are still in the pandemic 2 years later, and the pandemic has changed because the virus has changed, and it also has changed us. As you all know, there have been waves of mutations with ever-changing viruses that require new approaches, new thinking, and dealing with this, communicating with this, and especially communicating to the public how little we sometimes know about what is actually happening scientifically has been difficult. The virology, I believe, is the simpler part of it. What is much more difficult, what has been much more difficult as far as I can see, is understanding what happens in the patients. Why some patients succumb to the disease, others hardly feel more than a flu. This is not only genetic, although there's been tremendous progress in identifying genetic variations that predispose to or protect against severe disease. This has only partially, actually very little, advanced our knowledge to how the biology about the biology of the disease. On top of that came these new variants that pop up every 6 to 9 months. that spread because they're more infectious and that may probably be less deleterious to people. These new variants have a biology that is different, but we don't know yet exactly how different. Does Omicron infect lungs less severely than Delta? Probably. But what is the consequence? Does Omicron lead also to a long COVID syndrome? We don't know yet because it's too early. These kinds of questions, I think, are often not openly enough discussed, certainly not in the United States, and probably also not in Europe. And we need to be open, as I believe, as people who are connected to the healthcare system in some way or another, that there is an evolving understanding and that much of what's going on is simply beyond our current understanding, that we don't really know exactly what happens in the disease and exactly how the virus mutates and what the consequences of these mutations, of these new variants are. I think that science is always imperfect. It is always incomplete. That's why we continue to have a job. And we have to be able to try to communicate this, to try to convince the public that we as scientists are not some kind of priests who know everything and have very distinct and very definite prescriptions of how to deal with it. We need to communicate that we are basically building the plane, as they say, while we are flying it. And this is a process that requires, I believe, a little more humility also from the side of the scientists and associated entities. The other feature that I think is really important is the fact that vaccines are great. They are game changers. But we have to face the fact that the game hasn't changed yet. We do have vaccines, and it has been an enormous progress. Millions of people have been protected against developing severe disease, often against developing any disease or any infections. But still, it is not only recalcitrant anti-vaxxers that impede progress here. I cannot agree more with previous speakers that it is irrational to be against vaccines. And I've had many discussions with people about this because I meet a lot of people who are against vaccines, and their arguments are often irrational for me as a scientist. Moreover, we have vaccine mandates in many places in the United States, and I'm sure in Europe also, although probably not as much, and they're effective. They work. Nevertheless, the fact that COVID-19 changes, that we have new strains coming up, and I bet you Omicron won't be the last one, means that with the vaccines, we will always be a little behind. Even vaccines that are currently available are protective, clearly, against Omicron, but they're only partially protective. And we have to accept the fact, I think, that it may be possible that we will never be able to control COVID-19 as we, for example, control measles or rubella or some of the other classical infectious, highly infectious diseases for which there's effective vaccines. It will always be a little different. And I think we have to come out with that, and we have to communicate that, and we have to tell people what to expect. I also think we need different types of vaccines. Although the mRNA vaccines are unsurpassed in terms of how quickly they were developed and how well they work, it is really amazing. It's truly, as others have pointed out, a triumph of science and of biotechnology that these vaccines could be developed so quickly and that they have been demonstrated to be safe. Nevertheless, I think we need other types of vaccines for a number of reasons. For example, that some people react badly to the mRNA vaccines, that some people feel uncomfortable, and many other reasons. And in order to achieve that, we also again need to communicate to the public that there are choices, that there's a future, that these vaccines will never be 100% effective, but that they are actually useful, not only useful, that they're even essential for advancing the public health, for changing how we as our societies approach this devastating pandemic, and for a long-term dealing, for a long-term ability to deal with this pandemic and really turn it into an endemic situation where COVID-19 will always be around, but will be manageable. So to sum up here, I wanted to add to previous speakers who have, as I said, done such a better job than I could possibly do in summarizing what we know and how we might deal with it. The one feature that I feel as a scientist we fail at, which is communication, accepting the fact that there are limits and trying to engage people in helping us move forward. Thank you very much for your attention.
Thank you very much, Thomas Süthoff. And now we are ready for the final panel discussion of today on the lessons learned from COVID-19. This panel will be moderated by Thomas Seckeres, President of the Austrian Medical Association and the Medical Association of Vienna. He will discuss with our prior speakers, Alexander Biach, Leonid Eidelmann, Ottmar Kläuber, Stefan Ludwig, Heidi Stensmüren, Thomas Süthoff, and they will also be joined online by Kurt Wüthrich, Nobel Laureate in Chemistry. May I ask the panelists to come on the stage, please?
Hallo. Hallo. Ich setze mich in die Mitte. So a warm welcome to all of you. Thank you very much for joining via Zoom, and thank you very much for coming to Vienna and participating in this extremely high-ranking talk. Our topic is how we can fight together the virus, and I think that we have different ideas and different ways how to fight the virus. Maybe I can ask President Heidi Stensmüren from Sweden, who is president of the Swedish and the World Medical Association, about the Swedish way, and how do you think is the best way to cope with the virus, and how is it going on? So we all hope it's soon going to be over. We have various options which we didn't have in the past. We had this vaccine within an extremely short period of time, thanks to the mRNA technology. Sweden had a little bit a different way. I would like to hear your judgment whether it was better than in other countries or not, and how is it going to be in the future?
Thank you. Well, that's a small question. It's a huge question, actually.
Yes, I know.
I'll try to give a qualified answer. I don't think anyone can claim to have the best procedure, the best handling of the pandemic. We just don't know. It still puzzles us, this virus, and surprises us. Yes, Sweden did get a lot of attention and was considered to choose a different approach than many other countries. But when we look at it, it's not that much of a difference in many ways. But one of the strategies that Sweden pointed out was that, well, we looked at the consequences not only as a medical issue, but how it would affect the whole society. And as I said in my speech, closing schools, preventing children from attending school has huge social consequences and also economic consequences. And as we assumed this pandemic would be for a longer period, we knew that that consequence would be very immense. So we tried to keep children in school. I guess that was one issue. And it has proven to be long-term. But we also said that since it's going to be so, the duration is going to be long, the principles of social distances and the restrictions on the population must be quite simple. They must be easy to understand, and must try to make them from the start so that we don't have to adjust them many times over the time, and that has been fairly successful. But we failed miserably in preventing the spread to the elderly, because it did spread to the elderly. So, which is was very unfortunate. That was a short answer. I can elaborate more, I guess.
Thank you very much. Maybe we ask Israel how they are dealing with the virus. They were the first where vaccine was available. Not as many people got vaccinated as expected, but still the vaccination was extremely successful. And now you have high numbers of infected people, but low numbers in the hospital. So how is it going to be within the next weeks? And do you have any restrictions?
Thank you very much, Thomas. It's the situation in Israel, as I described, was a little bit different than in other countries, although during this pandemic, every country chose its own way. Because we have started very early with massive vaccination. And it looks like it helped very much. It's changed the situation in Israel. And also the decision about the booster was taken very early. Actually, no other country, as much as I know, decided about booster before that. And it also helped against the Delta variant. And you see, as every country, we had some periods with strict isolation, some periods with very limited measures. And nowadays, it's much easier than it was, and even for school children. Now there is no need to be isolated when there is some case in their class. We had it for a long period of time. And, you know, we always discuss it. No one is happy with any measures. I absolutely agree with Heidi that what lacked from the very beginning was the protection of elderly population. And I spoke about it at the beginning of 2020, that the elderly population should be particularly protected. But, you know, it's not very easy to do it because we are different than in Sweden. In Sweden, people live probably much in more distance than one of other. And we have very big families. And to isolate old people is not so easy. Even those who were isolated in nursing homes, they paid a very heavy price by decreasing of their everyday activity and by deprivation of relatives that couldn't come. So everyone understands that it's not a time to summarize all the lessons of these pandemics. But what is very important for politicians and to have leadership and will and decide what they want to achieve. If the calculation is according to the number of confirmed cases, the strategy is one way. If there is an aim, the goal is to decrease the number of dead people, the strategy is different. So definitely we will be in this uncertain situation for more weeks, but I believe that Omicron is a great opportunity to get herd immunity, and to end with this pandemic phase of this viral infection. Definitely we will have endemic cases, but I believe that in 1 month from now, we will be in the endemic situation and not pandemic, at least in Israel. Thank you.
Thank you very much. And we all hope that the pandemic situation will soon be over. How is the judgment of the virologist?
Well,
Professor Ludwig from the Center of Molecular Biology and Inflammation from the University of Münster.
Well, I think I can line up with the previous discussions that we are now in a critical phase of the pandemic, and with this Omicron variant, with regard to a population where a lot of people are vaccinated, not enough, it's true, but there are also recovered people which are immune, that we might be on the turning point to enter the endemic phase. The problem that I see as a virologist is that in the moment, due to these enormous amounts of infections, the sheer number of virus, the sheer mass of viruses, which is still existent on a global scale, of course, has the problem that new variants can arise. And this is something which we really have to prevent. So if the virus replicates on a low level in some susceptible people, we do not have this problem, especially with coronavirus, because coronaviruses are actually not so prone to produce variants because they have a proofreading function in their polymerase. So they are not as prone to changes as, for example, the flu or so. But it's the sheer number or the sheer amount of virus which is now in the world that leads to the generation of these variants. And although for the virus, it's better to be less pathogenic because it's not good for a virus to kill its hosts because then it cannot replicate anymore. And thereby these pandemic situations may enter in a less pathogenic variant and then endemic situation. This is not for granted. This cannot be predicted. And there may well be another more severe variant coming up in the future. And we really have to to have a look at that. Although I think, to be a bit more positive here, that the most likely scenario is really that we will enter an endemic phase.
But what would be the option? Lockdown, I think it's not an option at the moment.
I would not say that we should go back to these harsh measures. I think the key is here easy to be followed measures, like wearing masks and wearing them correctly. This is really key. We have seen that this helps a lot. And then a little bit of distancing, yes, that's also something which can be made. But there should not be, like in Germany, where we have these 17 states, and you have different rules in every state. That's not the way to deal with this. And that is also why a lot of trust has been lost into the governments. So some measures which are easy to follow, which can also be easily controlled. That is also something which can be done, and I think with that we can cope with the crisis.
Well, thank you so much. How is the situation in Switzerland? Professor Wüthrich, Nobel Prize laureate in chemistry from Switzerland.
Well, good morning. I am actually in California.
Sorry, but sometimes you're in Switzerland as well.
This happens, yes. I regret not to be in Vienna. I am not a medical doctor. This is why I preferred not to give an introductory speech. Now, should I take some 10 minutes to express some thoughts from my side at this point? I'm told that you got an extra 15 minutes on the panel for this, or is this a bad moment to come in with a general set of remarks?
Please.
That means, yes, go ahead. All right. Well, as I said, I'm not a medical doctor, so I'm really a bit shy to even open my mouth here. My background is, except for a couple of honorary degrees from medical schools and pre-medical studies as a sports student, my background is in chemistry and physics. And also, I was very actively involved in the bovine spongiform encephalopathy, BSE, or mad cow disease crisis, approximately a quarter century ago. And from those times, I have in particular very mixed feelings about the predictions of the outcome of pandemics and endemics. As a non-medic, I want to express my admiration and thanks to what in the United States is referred to as first responders in the medical profession. As a scientist, I notice with satisfaction that decades of curiosity-driven basic research are now getting known to a wide public. It is less than 30 years ago that the Nobel Prize in Chemistry was awarded for the invention of PCR, which is today in everybody's mind. So it was Carrie Mullis who was awarded the 1993 Nobel Prize in Chemistry for having invented PCR. Of course, we see in most direct ways that high efficiency in applying medical services has, in the time of the pandemic, become more shiny than the basic research and the innovations that are behind it. A striking example has already been referred to, which is that in Vienna, PCR tests are performed at the rate of 500,000 a day at the price of €6 per test, whereas in Switzerland, we are stuck with 100,000 tests per day at the rate of 88 Swiss francs per test. These are enormous differences, and it is amazing that we have such differences among small neighboring countries, even within Europe. It already shows that awards for achievements in the pandemic will go primarily to entrepreneurs rather than to those who established the basis. I want to refer to an achievement in my own field, in structural biology, where two of my friends at the Scripps Research Institute here in La Jolla, Andrew Ward and Ian Wilson, in 2015 determined that the spike protein of coronaviruses needs to be modified in order to serve as a target for efficient vaccines. And this is a so-called 2P variant of the spike protein. It has been patented. It has quite good effects on our department, but nobody talks about this basic achievement, and there would be no vaccine without having access to this 2P variant of the spike protein. What I then observe in the way things have been going on during the last 2 years, and I have observed this in Switzerland and in the United States, where I live, but also in China. I mean, when I came to the Vienna Congress in 2020, I arrived in Vienna 2 days after leaving China, which I left at the very last day that it was possible, because before the complete lockdown. And I'm in constant contact. I have about a dozen graduate students that I advise in Shanghai. And so I'm in virtually daily contact with them and know how things are going on down there. Now, when I watch what is happening, then I see a world full of reactions. There are very few actions. Whether or not that's good or bad, then the Chinese government definitely takes action. They want to follow a non-COVID politics. That's certainly action and not, well, it's a huge reaction, and I do not believe that we would want to live necessarily under these circumstances. I have not been back to China since January 2020 because of the difficulties of living under this regime. But let me mention a very particular example. We know nowadays that primarily transmission of COVID-19 goes through aerosols, not just through droplets, but through aerosols. Now, this has been known since the summer of 2020. Nobody has invested money in improving the air indoors in public places, in particular in schoolrooms. I intervened with the task force in Switzerland for more than 1.5 years now that something should be done about improving indoor air in public places. One of the results was that parents complained that using CO2 detectors to monitor the air quality in schoolrooms would psychologically affect the students and should therefore not be used. Not to speak of installing air purifiers. The same happens in California. We have here a huge political movement that says let them choose, meaning the school children should be able to choose whether or not to breathe in bad air or to wear a mask, or I suppose that Thomas Südhof is aware of this action as well. The next point I want to take up is communication and trust, which has been extensively discussed in the introductory talks. What has bothered me all the time is that the voice of the scientists did not go through. It is clear we have also heard from Dr. Piak about the difficulties of politicians to take in information from all sides. There are so many interest groups and so many conclusions over the last 2 years have been, well, I don't want to say erroneous, but let me call it differently. The scientific facts have been diluted by considerations on ethics, economics. I give you an example. I mean, in sports, for many months, only professional sports could be pursued. In the United States, in Switzerland, and in many other countries, whereas children were not allowed to play even outside and to pursue sports. I mean, such sort of decisions are simply very difficult to understand, and they do, of course, not help to increase trust in the population when it comes to whatever is communicated to them from the governments. And finally, I would like to comment, and I am afraid I'm repeating what has already been said. What possible long-term effects will we have? Well, the most important long-term effects will be what we missed in education. In addition, there will be a non-reversible trend of increased use of information theory and artificial intelligence, which in some countries will also be used to exert control over the population. Now, let me just comment briefly on the educational situation. In Switzerland, my grandsons missed school for 6 weeks in total. In California, schools were closed for 18 months, and it is a very slow process full of political discussions that the schools are now being reopened. And every week we see on television how a particular college, university, primary school changes its policy regarding the ways in which the children are supposed to return to school. In China, everything was down for 3 months. That means You see, the pandemic in China started, of all things, during the Spring Festival, during the week of the Spring Festival. And 500 million Chinese had traveled away from their workplace to places where they visited with relatives. Now, for 3 months, my students were not allowed to return to campus. They had to stay home. And after 3 months, things were more or less under control. And my students now work in the lab, and everything is more or less normal. However, there is always a possibility that there are 2 or 3 cases of COVID-19 in Shanghai, which is enough to close the campus for 3 weeks, so that nobody can get in and out of the campus. So we have these different systems which lead to different results at very different costs. The bad thing, especially what I see in California and probably in the United States altogether, to a lesser extent in Europe, is that the inequality between poor and rich parts of the population will be increased by the fact that education has been interrupted in such serious ways by the pandemic. These are some expressions of my thoughts that came up mostly by listening to the speakers in the introductory part to this panel. Thank you for giving me time to also bring in some of my comments.
Thank you so much for your thoughts and important input. Dr. Bier, what is your opinion, and in particular, how is the economic situation going to develop after this pandemic? Do we have a long-lasting problem in the future, or do you think we will overcome the whole situation? Dr. Bier is director of the Vienna Chamber of Commerce. Therefore, I asked him more economic questions and not the medical part.
Well, thank you. As I mentioned before, smarter heads than me can speak about the medical situation, but in fact, the medical situation infects the economical situation. And for example, I just took a look at the figures before. Closing down the businesses only in Vienna means that you lose about 2.1 billion euro for closing it for 6 weeks. So this is a real huge amount, and all efforts are worth and can be calculated in order to fight this pandemic situation, or in order to fight the lockdown situation, as also managed, this was only the business closing, but as managed before by Professor Wüttrich, closing schools is even worse in the situation at the moment closing it and in further development. So what we try to do out of the economical point of view is to avoid lockdowns, further lockdowns. There's a strong discussion at the moment running of reducing the points and the rules of control. It is some kind of, you have to be very careful in solving this situation. What we try to fulfill is we contribute to the formula for fighting the virus or the pandemic situation. This formula reads, being smaller than 1 means that if you can bring the effective reproduction figure of the virus below the level of 1, then the virus even finds not so many people getting infected. And there are 3 ways we try to to support from the point of Chamber of Commerce and the economy. The first one is vaccination is the key factor for reducing this production number, as shown before. The second one is to look at measures like the mask. Wearing the mask even lowers this reproduction number. Well, and the third one is where I mentioned before in my introductory note, where we put a lot of effort is into an effective PCR testing system, as we are very sure about the fact that finding the people which are infected and isolating them doesn't spread the virus by the others, so throughout the others. And there we put a lot of effort on this sector during the last summer holidays. During this time in Austria, the schools are closed. So we tried to prepare this testing system for all the pupils in Austria, whole Austria, in order to get them 2 or 3 times PCR tested, as we also realized that the reproduction number under the people of age 14, 15 is a very high one. So finding those which are infected helps to isolate the virus reproduction. And as I mentioned before, from the economical point of view, all this investment is worth being invested, as the alternative is to have another lockdown, which even costs more than the tens of the investment for medical measures.
Thank you so much. Maybe 2 sentences regarding the PCR tests. At the moment, it's a little bit more difficult to lower the number of infections because the incubation time is too short and it takes a few hours until you have the result. And maybe one word, that the price difference between Austria and Switzerland can be explained because we do pooling. 10 samples are being pooled. So I don't know whether you pool the samples in Switzerland. If not, then it might explain the difference between, because then we have 60 euros per one PCR test.
Are you addressing me with this
Yes,
question?
because you mentioned the difference
yes,
in price.
Of course, but I mean, the numbers that I give are for individual tests. And I think that these group tests, this pooling, is not efficient anymore at the present time because there are simply too many infections. So you either test individually or you forget about it.
Yeah, you're right, but what we did in the past was pooling of 10 samples, and that's one reason that we could offer the lower price and the high number. At the moment, the more positive samples you have, the more difficult the testing is, and you have to test again and open these pools. That makes it more difficult. But for a long period of time, we had only a few infections, and then the system worked fine. But
my information on these numbers of €6 per test in Vienna and 88 francs per test in Switzerland and 500,000 versus 100,000 per day. This information is from a Swiss newspaper of the day before yesterday. So that is the latest information that I have available.
No, your information is absolutely correct. So, yeah, we try to do our best in Vienna, and I think we managed the situation so far relatively well, and I hope that the upcoming weeks will be manageable as well. In particular, the hospitalization numbers in the world are not very high. The infection numbers are high, but they are not as many patients in the hospital as they used to be at the beginning or during the last virus variant. Maybe Ottmar Kläuber, the Secretary General of the World Medical Association, can tell us a little bit about different countries and how they cope with this challenge, virus challenge.
Now, Thomas, is it the testing challenge or the challenge with coping the virus and doing the vaccination what you would like to talk about?
Yeah, what are your experiences in the different countries? Of course, vaccination is a challenge as well. Yeah. And we see huge differences in the number of people being vaccinated, and how to cope with the whole situation.
Yeah, there is, in the first place, there is no winner. As Heidi said, it's very difficult to say that really there is one model that is an ideal model of testing or of vaccination and coping with the disease. Certainly some of the countries have been much better in rolling out the vaccination campaign than others. That is certainly true. On the other hand, there is no really ideal situation that we have seen. What we have is we have a big imbalance in equity, in access to vaccination, but also into other materials. And we have a huge north-south gradient in what means getting chances to survive the pandemic for either individuals or also the healthcare systems and the economies. And what we see currently is that poor countries, especially those of the global south, have it much harder to deal with the pandemic than we as the rich country up here in the north. But even in the countries in the north, I would say that we have been less than ideal in dealing with the pandemic. Think, for instance, of the pandemic preparedness. We have seen nearly no country in Europe and many other places in this world that really has been prepared to deal with the pandemic. There have been pandemic plans, which probably have been in some drawers, but a real preparation, which means exercising, which means detailing the preparedness down to each local public health system, to each local hospital, it has not happened. It has happened in some countries. There have been some in East Asia which have been prepared. Those which have learned their lessons from SARS and MERS some years ago, which have been much better. But for us, I think we have suffered a lot from the complacency in saying, well, pandemics happen somewhere else in the world, but not with us, unfortunately. So we didn't have the preparedness. But even then, when the pandemic hit us, it's not only that it's very difficult to predict what will happen in a pandemic. Unfortunately, we have not even been able to monitor the pandemic properly. Up to now, our numbers which we have between the different countries are not really comparable because we don't know what has been counted as what in which country. So what really was a death due to COVID or in connection with COVID, different from country to country? What have been the inclusion criteria to bring people into intensive care in countries has been very different from country to country. So, is there a country which has gone an ideal way? No, I'm sorry, at least I have not identified it so far. Have there been countries which have made huge mistakes in this? Yes, unfortunately, that has been the case, and many countries have missed the opportunities to do so. Have we seen political failures with that? Yes. I think I don't have to give you the names of the presidents of some countries which really have messed up the situation in the beginning. I think we all know that there have been countries which have been doing really, or had a very bad start because politicians were standing in the way to an optimal solution to that. But for us, all of us, there is no real reason to clap our shoulders. We could have done better, we could have been better prepared, we could have been fairer with the poor parts of this world. And I think these are the lessons which we have to learn in order to better in a next pandemic.
Thank you so much. Yes, you're right. If politicians are in trouble, then they ask the scientists to help them. That was at least my impression. Please, Professor Wüttrich.
Well, I think that we have here a very important point regarding communication and trust. Again, just a few days ago, statistics of the Children's Hospital in Zurich were published. 2 weeks ago, it was reported that by now 60% of the patients were COVID-19 patients. Now, the renewed analysis has shown that 70% of these children had been sent to the hospital for other reasons and were then found to be carriers of COVID-19 as well. And now in Switzerland, they are reassessing the records of all the hospitals to find out how many of the COVID patients that had been gone into the statistics had actually been admitted to the hospital for other reasons. And I think this sort of misinformation or poor information is simply eating away at the trust that the public has in those who are on television, in the newspapers, and so on, and try to influence the population in a positive way.
Thank you very much. Yes, you are right. I mean, we even tried to set up an international register, and to be honest, it didn't work. It would have helped us, especially at the beginning, a lot, but somehow the communication between the countries and with the EU didn't work as I hoped or we hoped. Professor Südhof, what do you think, which are the lessons we learned, and what do we have to do to cope with the situation now?
I'm not sure, excuse me, I'm not sure that I'm really in a position to speak to that, because others, like many of the members of this panel, are so much more experienced and so much more knowledgeable in the political side of things. And these are political questions. These are not scientific questions. How to deal with it, what to do. I can only say from my own personal experience, and this is very personal and has nothing to do with my job as a scientist, that living in the United States, as I have for almost 40 years now, over the last 2 years has been both a very positive and very negative experience. As was alluded to earlier, we used to have a president who advocated injecting bleach against COVID-19. Thankfully, that has passed. But there's still a huge dissension in this country about how to deal with the situation. A dissension and disruption, an enormous, you wouldn't call it discussion, it's just very, very, very acrimonious and very, very difficult to face, because after all, the current pandemic is a tragedy for thousands, hundreds of thousands of people, and we should be much more compassionate towards each other and not argue about things that in the end make no progress. On the positive side, I actually had good experiences with our local school system. The schools were basically only locked down for a couple of months here in at least where we live. There was Zooming of the kids from day one, which worked quite well given the circumstances. And locally, people were incredibly supportive and very compassionate. So there is this disconnect between the bigger country and people who don't even talk to each other, who think they're nuts of each other. There is no communication possible, at least in the United States, between certain segments of the population, which is a tragedy, which is awful. And then locally, there's actually quite a good effort. There's real help. There's tremendous support. And I wish, I'm not in a position to do anything here, but I wish there would be a way to translate the local spirit of cooperation that I've seen into a larger arena and how basically, certainly nationally, but also internationally. That's all I can say.
Well, thank you so much. Professor Eidelman.
Thank you very much, Thomas. I would like to stress the importance of science. The science is the main topic of this Congress. And in preparation for future pandemics, and I am afraid that we will see new pandemics in not so far future. And in specific questions, I will give you some examples. We don't understand why some patients develop cytokine storm, as was mentioned. Why some patients feel so bad from the very beginning, and why we have so many patients in the intensive care unit. But also, why those who were infected, some of them were infected 2 times, even 3 times during the pandemic. And why some that were exposed have not been infected. We have seen families with 12 children. And in this family, 2, maybe 3 members of family were infected, and some and others were not infected. So there is a huge work that should be done by scientists to give these answers as a preparation for future pandemics, and also to give the right way for politicians to contribute to preparations. I believe that is the most important scientific and political question. Thank you very much.
Thank you so much. President Stansmuren.
Yes, thank you. Well, I think there are some highlights to make before we proceed. And one is that we have a lot of other healthcare issues as well that we need to handle. And many of them have been put on hold. We have extended waiting lines for normal procedures, and we need to handle those as well. And we need to have an awareness that we don't have a backlog on cancer treatment, for example, and cancer diagnostics as well. So we spend a lot of attention on COVID, and we should, of course, but we need to see to that we can handle the other diseases as well, because they are just as important. And this is an awareness that we need to have. A positive side effects of this pandemic and the research and vaccination is maybe also the awareness of the importance of biodiversity and heterogeneity. We have learned from different scientific sections, you might say. We've also learned how and seen how important knowledge from biology and research in other fields is for medicine, which is positive and will hopefully make us be better able to have a sustainable society. And the last very good lesson I continue to bring up is the importance of stress testing the healthcare system. In financial system, we have stress tests because they are essential for society. If the financial system goes down, it will affect us a lot. Well, the same should be done with healthcare, and that's what happened in the pandemic. We were hit, and our capacity wasn't good enough, and we needed to scale up. And those who managed best in healthcare, they were able to scale very fast, move people from departments to another department, go away from single rooms to use like 4-bedroom, 8-bedroom. I opened up a section of 12-bedroom, just like that, over in some hours. Okay, if you have the capability to do that, you will be able to handle that better. And these are knowledges that we need to bring further and applicate on healthcare. So we should discuss the stress test healthcare system better. What will happen if we will have a 30% drop in hospital beds in a few days? What will happen, and what will we do? How will we act? And have plans for that. What will happen if we will quadruple the numbers of infectious diseases coming in the hospital, and make plans for that and say, okay, this is the scenario. What are we going to do? So we can learn from the financial sector and banking.
Thank you so
Be
much.
humble.
Yes, one of the arguments in the past was that the health system is important and the politicians shouldn't save money within the health system. Of course, we have a highly developed system in our countries, Germany, Switzerland, Austria, Sweden. And there are other countries where they don't have this highly developed system. And of course, in a situation like a pandemic, you can see the differences. And for the future, I hope that they are not going to save the money which they spend now. They should be aware of the benefits of a healthcare system at a high level with some capacities and the possibility to cope with situations like this. Also, we all didn't believe that anything like a pandemic can happen in our lifetime. So, who would like to comment? Please.
So I thank Professor Heidelmann for stressing the importance of science to solve the open questions which we still have for COVID-19. There is still a lot of questions we have. And one is, of course, whether there is a genetic susceptibility, why people get infected, why people get severely infected and others not, and so on. But if it comes, so this is important also for future pandemics, but when it comes to pandemic preparedness, I think, or I want to give here the discussion another twist, that we have to think even broader. And what I'm talking about is that COVID-19 started as a zoonotic disease. So this is a zoonotic virus. This came from animals and jumped to humans. And I think if we talk about pandemic preparedness, we really should have that in mind and have to ask the question how to cope with the zoonotic transmission. And this leads into the question whether one should not put on here a One Health approach, and whether this is something which has to be done to really be well prepared for a future pandemic, to really see everything as a whole. So human health, animal health, but also the health of the environment. This is all connected, and this is in a balance. And if you tip the balance in one of these areas, this will influence the others. And so this all has to be taken in mind. And I think, especially if it comes to zoonotic transmission, we have probably to enhance the monitoring in areas where this happens quite often. Because guess what would have happened if we would have been early enough to detect these transmission events? I don't know where it was, in the live animal markets or so, then we would have prevented this enormous disaster. So we really have to think pandemic preparedness at this global One Health scale and have to interlink the areas of human health, animal health, and also the environmental issue.
Maybe we don't need these markets anymore.
So?
Maybe we don't need these markets anymore. We shouldn't have this live animal market, something which we don't know in Europe. But
you can easily ask for that, but this is some cultural heritage of these countries. So you cannot simply take away their culture. This is also a critical point that has to be done very, very safely. So
yes, but we learned how small the world became. Anybody wants to comment?
Well, I think the One Health approach is more than just looking for the so-called wet markers, where you have wild animals which are being brought to the market, living and being dealt with in a very close space together with other food and other materials there. It is, of course, for us easy to say, well, let's give that up and let's stop that. But I completely agree with Professor. There is indeed a cultural heritage that we also have to respect and have to be careful with. But it also means that we ourselves have to look on how we deal with our environment. And let's, for instance, look at our farming, what we have here in Europe, in the European Union, which is highly depending on enormous amounts of antibiotics which are being used. So it's not only that we have to complain about others. I think we have enough reasons to look on our One Health environment here in Europe, how we deal with our environment, how we deal with our livestock, and things that have to be fixed. We pay a very high price for that by having, for instance, a very strong antimicrobial resistance. And I'm not sure that we also not have other problems which we have not yet understood and seen because of that. So it's not only a problem in Asia, it's also a problem here. And what we do is, of course, we multiply the problem by the very short speed of a very high speed of transportation, which we now have by our exchange between the different economies, by transporting one problem from one part of the earth immediately to another. A pandemic 100 years ago has taken 2 years to spread around the globe. It now takes 2 days.
Yes, absolutely. Thank you. Please, Dr. Bier.
Maybe one point, just a little bit missing, but I think very essential for dealing with pandemic, epidemic situations in future. We should focus more on training medical staff, nurses, and also the university students, because the lack of capacity was not only the bad or whatever in the situation of pandemic waves, it was also the lack of people being trained on this job, dealing and somehow working with the instruments around the intensive beds. So this should be also one focus, in addition to what you said before, in analyzing with the help of scientific research the reaction of the virus, the transmission process from animals to humans. But I think we should also focus on being prepared for further pandemic situations in training people, especially on this job.
Yeah, thank you. I remember in the past, before this pandemic, we were discussing a lot how many hospital beds can be reduced, which hospitals can be closed, how much money can be saved. And I hope that this will change now, and we will focus on the importance of the health system and the importance of not just the technical equipment and the number of beds, but also the education and the number of employees in the health system. Professor Eidelmann.
Thomas, I'm as optimistic as you are about the future investment in healthcare and education and science. Unfortunately, I'd like to bring to your attention the example of Israel. We had elections during one of the waves of epidemics, and healthcare was not an issue at all. No media, no politicians didn't mention healthcare in the middle of this pandemic. And I am afraid that people lost interest in health. They are tired. They are tired with all this news. And I'm very much afraid that after the pandemic, our healthcare will not be better prepared than it used to be before the pandemic. And our goal is to draw attention of the public to the science and the importance of the science, to trust the science and to trust the healthcare, to improve the healthcare. It goes together, and I believe it's for preparation for future challenges. It's the most important work that should be done. Thank you.
Thank you. Yes, you're absolutely right. However, I had the chance to visit your healthcare system, and I think it is one of the highly developed. So, but that doesn't mean that we should save money in the future and not to further develop it. Professor Süthoff.
Just wanted to add that in the United States, we have a bit of a problem here in that the healthcare system is insanely expensive. In terms of percentage of GMP, it's the highest percentage in the world, but at the same time, it is organized in a manner that the inequality in the system is enormous, and it's just not working. And so, to me, the notion that we should invest more in healthcare sounds like a great idea, but at least in the United States, there's a huge problem in the organization of the healthcare system itself, which is much more pressing than the actual amount of money that goes into it. We see it all the time. I mean, even people who are well insured, like myself, we struggle to get access. And that is even worse for poor people. And there's many of them here. So it's not directly pertinent to our discussion, but I can't help pointing out that, again, we have a political issue here that is really something that needs to be addressed. And I can only commend the Europeans for their fantastic healthcare system, which spends less money on getting better care, as far as I can see.
Thank you very much for this remark. I remember when I used to stay and live in the United States some 30 years ago, and obviously it didn't change much. Professor Wüthrich.
I am surprised that long COVID does not have a more important role in our discussion when we talk about outlook to the future. In this regard, I have been worried from the very start of the pandemic about the fact that patients lost taste and smell. And it appears that long COVID is largely related to neurological deficits. And there have been initial reports which have not been reviewed as yet. These are pre-review reports that measurable quantities of the virus have been detected in regions of the brain of patients. I would be interested to get some information from the medical profession represented here about this aspect of the whole situation.
Thank you for the question. Who would like, please? Heidi.
Yeah, I can. Thank you so much for your question. I was actually reflecting about it. So I'm chief of a department or theme in Karolinska University Hospital, and we also houses the ward or the outpatient ward for post-COVID. And as Sweden was hit quite badly in the first wave, we do have relatively a larger portion of post-COVID patients in Sweden and in Stockholm, where, well, I work. So we do have some experience. And as you mentioned, yes, we see fatigue, neurological effects on the patients, and also the shortage of breath and cardiac effects, rhythm disorders. So, and there's a number of symptoms, organ symptoms, but they vary a lot. And I've heard, you asked about the expression in the central nervous systems, and I've heard the same, but I don't have the scientific evidence for it here, so I can't specifically reply to that. But yes, we see it from so many organ systems that would be a logic explanation. It's long-term for many patients, and we do see good recovery on especially the lungs, the airways, deeper airways. But we also see a very long-lasting fatigue. So there's what we are trying to do is to group in undergroups of patients, and one clear group that we find the one with POTS as a and heart rhythm disorders. So we have classified that. So we try to make a fast track for those directly to cardiology, and we have different ways to treat those. But there is still a large portion where we are not sure, and we do not have a treatment yet. So we run multiple tests in my unit on this.
How many patients suffer from long COVID? I heard here up to 10%. Is this true or too much?
Well, yeah, exactly. It could be. We have in Stockholm 10,000 patients diagnosed, and well, on a population of 1.2 million. So, but I can't relate to. I've heard 10%, but for surely we don't know. The problem is many were not diagnosed in the first wave because we had such a backlog of diagnosing. So we don't actually know how many were sick in the first wave, and if is the Delta variant more plausible to give post-COVID than the Omicron, for example? We don't know yet. So we need more research and more funding in research for this. We've had quite a good funding grants for the research for this, and we have multiple groups, and we do have some articles on the way out, but I can't really say the results yet. But yes, there are some very interesting aspects coming up.
But it's a very challenging situation.
It is challenging. Yeah. So, and this came late. So we saw it early in the hospital, but we were only collecting data very broadly. And of course, the acute phase of very many sick patients requiring ICU care had to go first, life-saving procedures first. So it started a bit late, and we have, well, I said we have good funding, but of course we have like I don't know how many studies on COVID. It's very many studies in Karolinska going on COVID. Post-COVID, not that many, of course.
But it seems that the neurological problem is the main problem.
Yeah,
that's what I learned as well.
But we do see them in very many organ systems. So, but there is you're bringing up such an interesting issue there. I would just say. Maybe next year I will come back with some more answers.
Yes. We very much hope that next year we can be here in person and that this hall can be filled with people listening to us, and we don't have to communicate via Zoom. We will see. It is now the second year that we had a discussion here in a very similar setting in a pandemic situation. And I think we are all optimistic that next year we can manage to meet in person, maybe visit the Vienna Ball, which we used to. And I would like to thank you all for your input and your thoughts. And I wish you a good evening or a good morning to California. Thank you so much.
Thank you.
Okay. Bye-bye.
Bye-bye.
It was a pleasure.
Thank you.
Thank you very much for this very interesting discussion. Thank you also to our audience. Now we reach the end of the first day of the Vienna Congress. Have a pleasant evening, and we see you back here tomorrow at 9 a.m. Stay safe and good night.